Good morning, hi everyone, and welcome to the second day of the first Colombian Brain Health Congress. We hope you found yesterday's session informative and thought-provoking. Due to unexpected airport-related issues affecting one of our invited speakers, there may be changes to the scheduled program. We are closely monitoring the situation and will keep you informed of any adjustments. Thank you for your understanding. Today, we will begin our exploration of brain health with a focus on leadership, dementia, and genetics. Our first speaker this morning is Dr. Victor Balcour, who will discuss brain health leadership and global collaboration. Please, an applause for Dr. Balcour. Dr. Balcour is a professor of medicine at the University of California, San Francisco, with appointments in geriatrics and neurology. His research focuses on the neurological impact of HIV across the lifespan, including in pediatrics and aging populations. He leads international studies on cognitive disorders related to HIV in Southeast Asia and Africa and directs the UCSF HIV over 60 cohort. He also provides clinical care at UCSF Memory and Aging Center and serves on the executive committee and the AIDS Research Institute.
Muchas gracias. Voy a hablar en inglés. Thank you so much for having me here and inviting me to speak about leadership, international leadership, and dementia. I'm going to use the example of our Atlantic Fellows program, which may not surprise many of you. I realized, I've now been working as a doctor for over 30 years, and I've had to pay my salary through grants the entire time. The way the academia works in the United States, you either bring in your funding or you don't have a job. No one has what they call hard funding. So it really was a gift for me when somebody walked through our halls and said, we're thinking of putting together a training program around brain. We want it to be international. And so it was a pleasure to speak to this person and work with my boss, Bruce Miller, to establish what became the Global Brain Health Institute and the Atlantic Fellows for Equity in Brain Health. Now, it's been going on for 15 years. I'm going to tell you a little bit about our philosophy of leadership in an international stage. I want to tell you a little bit about the Atlantic Fellows program first, for those of you who do not know about it. But mostly, the idea that I hope you learn is that the best way to work internationally is to make sure that the people on the ground have the greatest voice. And we've been able to do that through the Atlantic Fellows program. So the Atlantic Fellows program is run at UCSF and Trinity College in Dublin. My boss, Bruce Miller, very much an educator, says that through the training of a new generation of leaders, we truly have a chance to transform our society, which is a pillar of what we try to do with this program. The man who made $8 billion and gave us a big chunk of it to run this program was really invested in people. I had one idea that never changed in my mind: you should use your wealth to help people. These two things came together to help us understand what we were going to develop. We felt that a residential program at one of our sites here at the University of California or Trinity College was really key. People needed to have a chance to breathe, to get away from their jobs, to go into another place. And for a lot of people, spending a year in the United States is a huge addition to your CV. So we really wanted to do that as little as we did not want to take people out of the context. And so it's very important for us that everybody has a fellow, has a mentor in their region here, and that they have a reason to come back and a job to come back to. We don't focus on dementia itself as a brain disease. We focus on the entire aspect of dealing with dementia. So our fellows learn about economics, politics, policy change, as well as neurology, and they get a lot of clinical experience. Some of our fellows even call the time they're with us a behavioral neurology training because they see so many cases. And a real goal was to network people together. So the training is a very small part of what we do, but we do have all this coursework, 2 hours a week, that folks go through. But more than that, they're doing a lot of clinical work. We decided to focus on Latin America in the first few years because we knew some fantastic leaders like Francisco Lopera, like Diana Matalana, Nilton Custodio, who you heard from yesterday, Ricardo Alegre. So many people had done so much groundwork that we felt it was ready to have the next generation of leaders move forward. So you can see 75 of our fellows to date have come from Latin America, and quite a few of them as well are from Colombia, including Nano, who I'll tell you a little bit in the story later. When the year he was with us, he was waking up at 3 a.m. to teach his new PhD classes here in Colombia. The level of dedication that our fellows have and how connected they are to changing things in their own society. But we have quite a few fellows here as well, Pablo, who you heard of yesterday. We don't accept only medical doctors. We accept people from any kind of walk of life. So we have artists, we have lawyers, we have folks that are in the social sciences. We've had people in business. It's really very divided. And even among those that are doctors, they're not all neurologists. We have a lot of psychiatrists. We have geriatrics, internal medicine, and so forth. What's really interesting about our fellows is that they may claim a discipline when they apply, but they're not in that discipline entirely when they join us. So some of the biggest policy moves we have made have been with neurologists who talk to the governments and change policies in countries. So there's a lot of overlap in this interconnectedness among our fellows. And in one of our first fellows, Eleanor Bayan, who's a neurologist out of Paris, we thought she'd go back and work as a neurologist, but no, she did. Of course, she has to pay her salary. She made films for kids to teach them about brain health, and those are now translated into many countries. They're available. In fact, I was in Chile a couple of months ago, and somebody was talking about that, a poster of this My Brain Roby that's being done in Argentina. So people really do cross quite a bit. We lead with values. These are our values that our fellows learn about. And there's a lot of activity that they do in our center that builds leadership and community engagement. But what I really wanted to talk to you about is this concept of accompaniment. And I don't think I have a slide about accompaniment, so I'll tell you what the word is, and if I have a slide on it, then you'll get it early. Accompaniment is a word, I guess it does translate into Spanish, somebody told me recently, but it's a word that was made rather famous by Paul Farmer, who ran Partners in Health before his untimely death a couple years ago. And Paul Farmer talks about accompaniment as walking the walk with the people that you're with. So for us, it's about staying with our fellows and sharing and working with them on their journey. So we work as hard as we can to avoid what I call a transactional relationship in favor of a transformative relationship. We're not a degree granting institution. So you come and spend 12 months with us, you don't get a degree. It turned out that that was a little bit of a problem because people in Latin America needed to have a certificate of some type. So we now do have a formal certificate that's approved by the graduate division. But it's not for us to provide the answers for the fellows that come with us. And you'll see on a slide that I have coming up that the key is that we develop their strategies, not that they develop our strategies, right? So we have a strategy which is to accompany and support the fellows. And their strategies have to be at the center of change. Those will allow us to have transformative and sustainable solutions. So we think about that in all the work that we do. And here is Paul Farmer's quote: "To accompany someone is to go somewhere with him or her, to break bread together, to be present on the journey from the beginning to the end." So for us, it's about bringing people in, and at the point that they become a fellow, they become part of the family. And we listen to their answers. I was telling somebody yesterday, sometimes the solutions that they come up sound so crazy, we don't know whether we can do them, but they are informed by the local community, and you can see that we've had a tremendous amount of success. In San Francisco, it's important that we walk the walk as much as we talk the talk, so we've really invested in community engagement because we think that that is critical to advancing the dementia narrative around the world. So we want our fellows to see what's going on. So these are some examples. Sergio Lanata is a Peruvian neurologist that works with us, who does a lot of our leadership around the community engagement. And one of the most transformative pieces of what we were able to do is meet with Maria Carrillo, who is in the Alzheimer's Association Chief Scientific Officer in Cuba when we were starting to plan this work in 2015, and asked for her partnership. And she agreed to fund 2/3 of a pilot program. We decided that for our fellows to be successful, they needed to return to their country with money and a project. So all of our fellows learn how to write for money. They have to write a grant during the year. In fact, if you see any of them, you can talk to Pablo. He's got a deadline next Friday. It's very stressful for them to write these grants, but they learn how to write in written form, to ask for money, to describe something that's incredibly feasible, impactful, will change their lives. And we end up funding 70% of these. The Alzheimer's Association funds 1/3 of what we fund. These are 2 examples of some of our pilots, including Yared, who's doing work with our cognitive testing battery. So this effort has now funded, I forget the number, perhaps it's in the slide deck, but it's funded grants now in 40 countries around the world. This is an example of a grant. I'm working on an R01 right now with Elisa Rezende to expand this because she's gotten additional money from the Fogarty Center for R21. Now we're trying to get it into an R01. But she works in an area of rural Brazil where illiteracy rates among elders is very high. And the government is trying to provide literacy training, and she wants to use that as a natural experiment to determine if it can help prevent dementia. So far, she's shown that the intervention provides increased connectivity through the hippocampus, and now we're moving on to do a clinical trial. Lingani is the only neuropsychologist in Botswana. She's very interested in traumatic head injury, and she developed a protocol for post-hospitalization for people coming with traumatic brain injury. Surprising to me, the #1 cause of traumatic brain injury, I've traveled all over the world. It's almost always car accidents, mopeds, particularly the motorcycles. In Botswana, it's gender-based violence. So around the world, you need to cater things specifically for the environment that you're working in, and that's what she's done. Nahuel has trained psychiatrists to recognize frontal temporal dementia. If somebody comes in with psychiatric illness at a younger age, you should think about the possibility this could be a neurodegenerative process. Marta is Ecuadorian and has trained primary care doctors across Ecuador on the detection of dementia in her community and early treatments for that. So this is the slide that I didn't know if I'd put in. All in all, we funded $4.1 million worth of grants in 45 countries. 163 of these pilots have been funded. And together, those fellows have now raised almost $120 million for the work that they're doing. And I want to spend a little bit of time telling you what that looks like. We are collaborating for a lot of this work. You can see the circles here are mostly grants that are written with our faculty, UCSF, and fellows. The fellows take the lead, but our faculty often will co-lead with them. That helps us, we think, get through some of the review processes in the United States. But the Red Dot Consortium's one that's really linked throughout Latin America. And you can see the little circle in Colombia also has a dark blue. That's the D43, which is why we're here today. We also just got funding to do capacity building in Ethiopia and Kenya. We have quite a bit of work going on with bilingualism and English as a second language in Asia. I'll tell you just quickly about a few of these. I don't want to run over too much on time. But the Red Dot Consortium has now enrolled over 5,000 people across 6 countries in Latin America, and we're waiting for a possible renewal that scored well, and it's under review today at Council, so hopefully it'll do well. These are the sites that we enroll across Latin America. I should go back and tell you it's led by Agustin Ibanez, who you heard from yesterday. He's one of our Atlantic Fellows in an earlier class. They're starting to be able to define some of the genetics that's occurring in this cohort across Latin America. Some of this led by Juliana, who some of you know, and Stephanie Pines Guerrero, who is a fellow from Mexico, who has done a lot of the work on the social determinants of health. Sebastian Monglier is the lead author on the brain gap, the brain age gap that you heard about yesterday, showing that in Latin America, people's brains are aging more rapidly than they should, using this technique. And Sebastian as well was an Atlantic Fellow working with us. And Nano has looked at disparities here in Colombia, across actually the whole Red Dot Consortium, showing that in fact, these disparities drive dementia more than you would expect for if you use data that were from the United States, where cardiovascular disease might be more important. He also now has a grant with Michael Corley, who apparently is stuck in Panama, but coming down to be with you this afternoon, looking at epigenetics in this cohort. And this is an NIH R01 grant. You heard yesterday from Adolfo Garcia, who's also Atlantic Fellow, who's worked with Marilu Gono-Timpini, who studies language. I'm sorry, Adolfo came to us and said, Victor, what I really want to learn is how to write a grant. I want to write an R01. So we worked with him the entire year, and he got funded to do this work developing this TEL app that you learned about yesterday that allows you to do speech recognition. And by using parts of speech that I never would think about, granularity, neighborhood, length, he can use machine learning and do a remarkable job detecting dementia very early on. So you heard about his work yesterday, and I won't spend much time talking about it. We're here today because we got a D43 grant. Nano spent time when he was with us teaching the students here. It was the first year that the PhD program was in place, and he wanted to find funding sources to keep this program going. So that it took us 2.5 years, but we did eventually get a D43 grant that funds the seed projects. We'll hear about those this afternoon from some of the students, but also is able to provide some tuition to some students as well as stipends. It's a start. We need to raise more money and find other ways to keep this going, but it's a start to really supporting Nano's idea, which was we need a neuroscience PhD program in Colombia, and he had started it, and we're supporting it. Boonlead T has done a lot of work with language recognition. She's developed language tests for primary progressive aphasia in Chinese populations. So language is critically important as you do some of these tests because you're looking at failures and how people do grammar, for example, or what their word choice is. So it needs to be adapted across different populations. And she's done a lot of this work. She's working with another one of her fellows in Turkey, Didim, to do the same work there. So the network is clearly working. She's also working on a bilingualism. Is it good for your health? I certainly hope so. I've spent the last 5 years learning Spanish, and hopefully at some point I'll be able to give a talk in Spanish, and I hope my brain will last longer. So she's working on that type of work now. And you heard about Adolfo's Include network yesterday, which is now engaging people, including Boonlead and other fellows around the world. We have some work starting in India, where we're trying to look at genetics. Fahim is a neurologist working at one of the major hospitals in India, and he's enrolled almost 300 people with frontotemporal dementia. He doesn't have the genetics to do the work. So we're seeding that work now and trying to get him funding through other sources. Our partnership with the Alzheimer's Association was so successful, we decided to go into a new project where now we are doing some capacity building. 2 years ago, we funded our first 2 capacity building grants. There are 5-year grants, $250,000 a year. For 5 years, it's a lot of money for some of these places. We invested in one site in Mbarara, Uganda, very rural Uganda, doing amazing work with colleagues here in the United States, and now they're pivoting to do dementia work for the very first time. This one center is the second most successful center in the world for getting NIH K43 grants, which is a big statement. These are the training grants that come out of the NIH, so we're very hopeful for that group. As some of you may have heard about the brain bank training in Sao Paulo. There was a session last month led by Claudia Suumoto in the brain bank there. We've invested in that. They're the other recipient of one of our capacity building grants, develop brain banking expertise here across Latin America. And just 3 weeks ago, we launched Bridge Africa, which is a grant funded by the NIH through a UG3 mechanism, where we hope to build centers of excellence for dementia in Kenya and Ethiopia. And this is led by 2 fellows, Yared, who's a neurologist in Addis Ababa, and Chi, who is a bioneuroscientist, a biospecimen neuroscientist. The lead for the site in Kenya is also an Atlantic Fellow, Wambui, who has a master's level training in dementia. So it's really driven by our fellows in the field. And then we launched the first worldwide finger study in Africa last December. This will be done in 2 countries One is led by our fellow Chi in Kenya. The other one led by our fellow Doyen in Nigeria. So a lot of good things happening. We realized as we started to work in Africa a bit more, and I hope somebody will raise their hand if I'm getting way beyond my time, but we decided to work in Africa a lot more in the next 5 years. And what we found is they don't have cognitive assessment tools across Africa. People are using screening tools for the work that they're trying to understand diseases and changes in disease over time. So our fellows got together and created a battery we call the harmonized, multi-domain harmonized cognitive assessment battery. And we seeded that with the GBHI funds. It's now running in Arab-speaking Israel, which you heard about yesterday with Rafi, who's a fellow of ours, but also in Kenya, Ethiopia, and Botswana. And we're hoping to expand it into Cameroon, Ghana, Togo, Zambia, Tanzania, Uganda in the next year. This harmonized battery uses a lot of tests from other batteries, but also a lot of cognitive tests. And this is led by 2 neuropsychologists on our faculty. I'm going to close by just telling you a few things that I've learned over the years. I've been working internationally for 30 years. In 1996, some of you weren't born, I decided to drop myself in the middle of what was previously apartheid South Africa and work as a doctor in an area that didn't have resources. I haven't stopped working internationally since that time, and I've learned lessons along the way. Certainly, one lesson was I wasn't a very good doctor in post-apartheid South Africa, but I certainly learned a whole lot about what we can do, what we can't do. One of the big issues when people engage in global health from regions that have resources is that they have incredibly good intentions, good ideas that they think will work, and a level of sustainability that's embarrassingly low for the work that is being done. For example, we see lots of cognitive tests being done around the world. People have good intentions, they pull tests, have people employ them, and we end up with really terrible data about the frequency of disease. A lot of times even population sampling is ignored entirely. So we have what really would be a frequency in a biased sample that we see published as a prevalence in a country. It's really critically important that we get our hands dirty and join folks in these regions to not only provide what we think are some of the better approaches to doing this, but to listen and understand what the opportunities are and where you can move forward. In many places, these might not be the right questions to answer. There are other urgent questions to answer, and we have to listen and find those and then try to pivot ourselves to get into that. So many times these research ideas come from the global north, and they don't last. People want the money, they want the privilege, they want to participate in research, so it's so easy to find individuals who are willing to do this work. But it's not a sustainable solution because it's not what they wanted to do in their lives. It's very important to find out what individuals want to do in their lives and then match that with what we think some of the benefits might be in the region. So although a lot of these ideas have been well-meaning, global health research has really a terrible long-term experience that we have an opportunity to turn the clock and look at it in a different way. It's really important to have partnership early on. You don't really want to bring somebody on after the grant's written. We hear this a lot about this movement in Europe around patient participant voice, where people say, I'm going to do all these things and then I'll bring a patient in to give voice to it. Well, if you're bringing somebody in that late, they really aren't giving voice to what you're doing. And we see this a lot in global health, that we have great ideas. I am an expert in this. I want to study it in different populations. I think we're going to find great things. It's really important that the people you're working with also want those great things. And then this was written before the Trump administration, but inconsistent funding streams is really a tremendous challenge that we're having. And it really does make work difficult. The way we get funded is that we are answering a particular research question that is critical to people who review them at our NIH. Those are not always the same questions that are important and critical here. And so we can sometimes get moved into a different direction. Even in the United States, we can get moved into different directions. But it's a real barrier to moving forward. We may join with folks in a country to answer a particular question, and once the question's answered, leave. And that's a huge challenge with our work. So as I said before, we lead with our fellows strategies. So the fellows come to us sometimes with the most harebrained ideas you can imagine. And we work through them. We think about the feasibility. We think about the possibility. We think about the partners. We bring in a local mentor. We try to brainstorm about the feasibility. Sometimes these disappear. The ideas the fellows decide are not the right ones, but ultimately they're driving the strategy. Every single grant that I talked about was not a grant that anyone at UCSF wanted. It was a grant that a fellow wanted, but we had the expertise to match and move it forward. So thank you very much for your attention on this.
Thank you for your presentation, Dr. Balcour. I would like to ask if anyone has a question in the public. We have short time for questions.
So thank you very much for that wonderful presentation. And I was interested in the last comments that you made regarding the balance between the global and the local priorities. So the question would be, when directing research on global health within the field of cognitive disorder, how do you balance the global concerns with the local priorities?
Well, for the first time in my life, I have to worry less about the global priorities because we have a funding stream through this philanthropic gift. So I guess I'm protected a little bit from that. When our fellows come to us with ideas, I really try to vet the likelihood that we're going to be able to find funding streams for that. So that might be where it comes in the most. I was just talking to David about work in HIV. HIV and cognition, I think, is not likely to be having great funding streams moving forward. So when fellows come to me and say, I want to study HIV, I often have them understand a little bit more about how difficult it might be, because that's the global piece to it. But they're the ones that are coming with a question. And we don't, I should be a little bit cautious in saying that I'm protected from it, because we as an institute have focused on training people, not funding programs. Everything that you saw here, with few exceptions, are funded by external partners, including the Alzheimer's Association. We do seed things from here to there when we find gaps that we can't immediately fill because we need to have feasibility data and so forth, but mostly we're not. So we do have to think about the global agenda in that situation. And we think about who's funding and what are they funding. And it changes all the time. And then we just provide that information and allow the fellows to understand what they want to do in the research space, I should say. I didn't spend as much time, this program today is really about research. I didn't spend as much time talking about how we fund our arts. Those are even more challenging to fund, but we do. We are able to find funders to get concerts, to make movies. We've made multiple movies now. There are funders out there that want to do that in advocacy and stigma, particularly. Yeah. Thanks for the question.
I would like to ask you, how do you sort this recent situation, like politics, like crashing into science right now? Like how do you feel that?
Well, it's a tough question to answer. What I tend to pivot to immediately is moving away from a U.S.-centric view of the world. Because I think we're reacting to a very US-centric set of changes, whereas it's not the reality in places that I see around the world. So I think that would be the very first thing. I do not think the world is moving away from science. I do not think most of the people in the world, I don't think most of the people in the United States are moving away from science. So how do I work with that? I don't know. I struggle with it. I held it for a long time. We now are having open conversations with our fellows because it's a reality, but initially we didn't. Advocacy, a little bit of waiting. Things are going to settle out, I think, in this. And traveling and seeing how much science is important in other places of the world, because that's the reality.
Hello, doctor. I have two questions, and the second one is like a second part of the one that my friend just asked. So the first one is if some of your fellows have proposed projects on more basic research. I'm asking this because the projects that I know personally here from the fellows that I know are more applied, and the examples you showed us as well. But I know a lot of people who work in more basic research, and I'm wondering if they could somehow collaborate as well.
Yeah.
And so that's the first question. And the second question is, if you think there's going to be any impact in the future fellowships in the United States, given that you are bringing people and the current political situation is not very friendly with immigrants.
Well, those are two very different questions. The first question I will say is we were driven in 2015 by the fact that dementia will be a traumatic issue across low and middle income countries and consume economies, and that data were coming out that this is modifiable. So we decided that the limited funds that we have, we would focus on clinically focused work and really preventions. And so a lot of the work that we're doing and a lot of the people that come in are focused on preventions. I actually didn't give many examples of it, but that's where most of our places are. So that's the first question. The second question is a terrible issue. I have emails right now about how we will run our program next year. This is live, maybe I shouldn't say it, but we did something that embarrasses me entirely, but we took the word equity out of our program just to give letters of offer, because it is completely in our program. It's not coming out in any way whatsoever. It's on our website. But these letters have to go to our State Department to be admitted into our country. And we worried that if people were being admitted into our country for an equity program, we'd have visa issues. So we changed it this year. So I'm very worried about that. I don't know how it's going to work. I hear there are lawsuits now for various groups that are being denied entry into the United States for visas. At the same time, I'm optimistic. I think we're bigger than one administration. I think we'll get over it. We may do remote work, but we really think that the residence experience is critical. Yeah. Thanks so much, everyone. Thank you.
Thank you, Dr. Bakur. We are honored to have you as the director of the GBHI.
Thank you again, Dr. Gloria Patricia Cardona Gomez, who will present lipids as a biological indicator of brain impairment. Dr. Gloria Patricia Cardona Gomez is a professor at the Faculty of Medicine of the University of Antioquia and a researcher in the Group of Neuroscience of Antioquia, specializing in cellular and molecular neurobiology. She earned her PhD from the Universidad Autónoma de Madrid at the Cajal Institute, where she studied neuroplasticity and neuroprotection. Her research focuses on neurodegeneration, gene and cell therapy, and the role of lipids as biomarkers in Alzheimer's disease and related dementias.
Good morning, everyone. I am very happy to be here. Thank you very much to Dr. Hernando Santamaria for inviting me to be here in this significant event. Okay, we are going to do a change to this topic. Check. Okay, today I will share with you our research about the lipids as biological indicators of brain impairment. I invite you to think into the cell, think between the cells, think in the relationship between the brain and the rest of the body, the brain and peripheral relationship. Trying to understand what happened with the brain and how may give messages to the rest of the body about how it's changing, how it's improving, how it's healthy, or how it's going to the unhealthy state. When we think in that, we must... Todo desde aquí. Excuse me. ¿Tú tienes? Ah, sí, creo que sí. Yo me muevo. Okay, thank you. Excuse me, the pointer. Pero no da. Mira. Ah, ya, ahora sí, perdona. Okay, sorry. When we think in the cells, and specifically the cell components of brain, we must then, in our mind, the neurons, obviously, and also other neural cells, glial cells, in general, oligodendrocytes, astrocytes, microglia, of course, also vessels. But we must think that all the cells have constructed is conformed by macromolecules as proteins, not only proteins, because the focus of the scientist community is in proteins, but also obviously in nucleic acids, but lipids has a crucial role and has been a little forget it. When we have the healthy aging, that's continue working very well. That's continue communicating, transmitting, connecting, degrading, etc. But depending on lifestyle or genetic background, that could be changed. And we have alterations or the membranes that are the main constitution of that kind of cells. Sorry. Okay, right now. When we continue with the healthy brain, the interaction of these cells in the neurovascular system, in the blood barrier, all those are communicating the brain and peripheria. And when we think in neurodegeneration, we forget how, or forget no, we have the study more or less how it's in the beginning, how was working, and what part of this information is going to the peripheria. This is slowly progressing to the degeneration when we have unhealthy lifestyles, but also depending on the genetic background, that we have the susceptibility to develop any kind of neurodegenerative disease, between others chronic disease. And when we think in the time as an intrinsic factor playing in the brain function outcome, I don't know why, our community had focused during the several years ago, mainly in the final stage, mainly when we have neurons, astrocytes, microglia affected, or when we, without cognitive impairment, at least a clinical evidence, but we have some information by biomarkers, imaging, or biofluid markers. We have information of the middle stage with memory loss, speech difficulties, start with this dysfunction, and after moderate stage, severe stage, we are in a moment that is late. It's late for prevention, it's late for a very recovering of this. ¿No está funcionando? Gracias. ¿Me han escuchado? Okay. Okay, vale. Okay. When we check what happened with the brain has tangles, have amyloids. We have this kind of disorders. And the early biomarkers that are going into the biofluids, as the CSF, cerebrospinal fluid, or in the plasma, serum, the p-tau 231 or p-tau 217, is because we have aggregated proteins in the brain. It's because have occurred a lot of events, of biological events in the brain, several years ago. But how we study, how understand by the periphery that these alterations started. We know, the community knows, or have studied, that some genes alterations have involved and have hyperpenetrance, but others have protections, of the recently published the cry chart mutation in APOE3, and several genes are involved with predisposition, susceptibility to develop alterations of the brain, mainly focusing neurodegeneration in that case, type Alzheimer. But interestingly, even the hyperpenetrance genes and some predisposition genes are related with vesicles, are related with lipids, are related with the organelles enriched in lipids. It's talking about transport, it's talking about degradation rate, communication between cells, and we have not paid attention so much. When we think in the risk factors related with the lipids, in that unhealthy aging and neurodegeneration, and obviously we have a 5% of risk, genetic risk. These genes are associated with lipid trafficking, APOE, a transporter of lipids, and vesicles, in the membranes system are involved there, lipid synthesis and sensors. But when we revise the risks related with the Alzheimer's disease, sporadic Alzheimer's disease, we have other peripheral lipid modifiers related with metabolic disorders, obesity, diabetes, vascular factor, hypertension, high cholesterol. And recently, how yesterday was talking here, the environment, the contamination, the smoke, the infections, how predispose the lipid metabolisms. But when we analyze or study the lipid metabolisms, we know general things. We know that it's important as sources to produce energy. We know that depending on that, the proteins can go through the lipid rafts, navigate into the cells, and to have contact with another proteins and produce events, different kind of the cellular response. But we have also the land cycle. I present to you the land cycle. It's important in the communication between the brain and periphery. We have autophagy. This is more famous events, cellular events that degrade and is dependent of the energetic balance in the brain. The metabolism status is dependent of autophagy efficiency. But we have forgotten a little bit how the mitochondria, endoplasmic reticulum, Golgi's apparatus, and the vesicles is working, are communicating between them. When we try to think in the where is working does lipid metabolisms, our first idea is neurons. Yeah, the neurons are the kin of the central nervous system. And we know that the neurons depend on the cholesterol given by the astrocytes in stretch communication with the microglia, but at the time with the oligodendrocytes. Depending on the stage of the oligodendrocytes, the myelinization, the connectivity, the strength of the communication between cells, the efficiency to degrade rest of the detritus, debris in this case, that's worked very well. But if we share that system of the lot of toxins or a lot of the metabolic products that are not really good, that does system work very well, start to have problems slowly. When we must study biofluid markers in an early stage for primary prevention and therapeutic approach. When? Because the early that we know, the earliest is 25, that we detect amyloid by PET amyloid, by PET. But before to this state, what happened at the brain? How the neuron, how the microglia, the astrocytes, the endothelial cells get signals to the periphery and say, hey, I am changing. Then this is our hypothesis that had working several years ago. We consider that there is an initial metabolic disbalance of the components of the neurovascular unit impacting the homeostasis, which release lipids to the periphery as early biomarkers and prodromic of cognitive impairment. I mean, depending of all these endoexosomes, how this endolysosome system does work very well. But those are into the neurons, into the astrocytes, oligodendrocytes, microglia, and give information to the basal goal that we can detect. but the problem is how so sensible to detect the minimal amount into the blood. For several years in neurobiology area of the group of neuroscience of Antioquia, GNA, we have worked in tissues, in vitro cultures, in neurons, astrocytes, microglia, and we understand that if we blocks, we change the environment by gene therapy, cellular or pharmacological therapies, we can partially recover the function of different kinds of cells, even impact on the cognitive performance in mice model of Alzheimer's disease, for example, that we have in University of Antioquia. And we test the gene therapy, we can move the animals model of the Alzheimer's disease to the healthy cloud, yeah, to the healthy cluster, okay, and improve the memory and improve the learning and memory skills. But it's not enough, because here in Colombia to do the translational medicine is very difficult, for not saying impossible until now. Then we start in collaboration with the Neurobank in the group of neuroscience with Dr. Andres Villegas, and obviously Dr. Francisco Lopera is into all this work, house inspiration and support. We start to study the postmortem brains, and we because we didn't understand why, if we blocks some mechanisms into the autophagy related with lipids, stop the recovery of the animals. And all the answers indicated that the lipids was a very crucial in the recovery or in the disease. Then we continue, we focus in human tissues, in human samples, and since right now I'm going to focus on that kind of advancing that we have. Okay, in the frontal cortex, we study how the lipids change. The more important to mention here is that when here say dementia, we have in those cases of CADASIL, vascular dementia, sporadic Alzheimer's disease, and familiar Alzheimer's disease. And all of them, with this focus, with this topic, specifically with this kind of analysis, clustering at the same cluster, yeah? Really different to the healthy. And highlighting some lipids and composition of that lipids. When we check, this is a small evidence about this work. When we check, in addition to the gray matter, the white matter was changing too, and highlighting others and the same lipids. We go a little deeper to understand what happened, and FAT and SAD has the same disorders or enzymes related with dementia, with Alzheimer's, but indicating the mitochondrial dysfunction, indicating pro-inflammatory phospholipids were participating in this impairment. We, in that moment, we did a summary of the observations when we could see that the gray matter and CSF has a very similarities. If we try to understand how works the brain, it's obvious why the gray matter and the CSF has a very similar changing in a different way, in this case, inverse way, that white matter. But was not enough. Was not enough because it's postmortem. Postmortem say the late stage that we cannot do nothing. Then with the R21 project from the NIH, from NIA, National Institute on Aging, in collaboration in the beginning with Dr. Francisco Lopera, and continue working with Dr. David Aguillon and some medical students. We are focused in a core. That's a 313. We start with a little bit more, but some exclusions. We focus on the 313 cases and analyze it by Nelson Galvis, by the sex, female, males, by the carriers, was blind. We say to the people in charge of administrate the samples, we need carrier, not carrier, with this range of age, children, adolescents, young adults, and older people, and give blinded the group A in group B. Classified by age, classified by clinical status, and others, education, other socio-demographic analysis. Juan Pablo, I can see Juan Pablo right here, helped to organize this too. And with support of Ken Kosick too, we phenotypified that population. What kind of APOE has that population? 2, 3, homozygosis, heterozygosis, etc. And we study in those cases as a part of the rich sources of the Neurobank, Biobank of the GNA in University of Antioquia, we could have access to the samples of people very young, 6 years old, females and males. And we analyzed the serum in this population. And we, in collaboration with Estela Aria Gomez, that was several years, 20 years ago, 20 years in Colombia, and right now in CEIB, in CSIC in Madrid, we analyzed the lipidome in a high resolution mass spec. And what's expected about that lipidome in the serum of people, carrier, not carrier, males, females, children, adolescents. Okay, then I'm going to share with you that's a part of this information. This is finally the preparation for submit very soon. But we expect that the most of people, all of us, go to the physician and say, okay, cholesterol profile, triglycerides, etc. We think in something like that, and analyze. We know that there are lipoproteins that carry lipids and going lipids from a cell to other cell, etc. When we try to read the information of neutral lipids, cholesterol, free cholesterol, cholesterol ester, and triacylglyceride, etc., we didn't find difference here. This overlapping group A and group B, 96%. And we check this population is in FAD cases, familiar Alzheimer's disease, and this is population of SAD cases, sporadic Alzheimer's disease from Columbia University study. And we compare It's a little different because the blue one is the blue cluster is the healthy people, and the red one is Alzheimer people, but are overlapping a lot. And then we say this is the same, more or less the same, yeah. Then the neutral lipids say a little bit about what is happening and how the gene, the alteration in the gene, specifically in presenilin 1 280A, is changing the lipids metabolisms. But cholesterol, free cholesterol, triacylglycerides are plant form, for the cells can function. The cells may not lost the cholesterol because die. Then does change a little bit, very a little bit. That is not significant statistically, but the statistics say so only a part of the information or highlight a part. Then we study other phospholipids, sphingolipids, glycolipids. What kind of information we find? This follow slides show the profile of lipids from serum of fat cases in different range of age that I'm going to show. That is, lipidome from serum and fat cases at 6 to 12 years old, females, males, and together. Here I'm going to show you together because we have information, very interesting information, depending on if is female or male. Okay, say that the serum is going out to the serum a lot of the sphingolipids in this age. That we can see the high here. We have overlapping some part of this information between healthy and unhealthy, or unhealthy, no. It's healthy people, it's young people, it's children's, but carrier and no carrier. And a prediction of 86% with this profile by the using machine learning, Dr. Julian Arias advise and follow us about this analysis. Okay, what is the information saying? That where is all these kind of lipids are in the cell scenario that I said you before. In the reticulum, in the Golgi, in the membrane system, in the lysosome, in the extravasicles. Okay. What happened at 13, 19 years old? Almost nothing. Why? 13, 19 years old is mainly by volcanoplot. Only significant here a reduction of monoacylglyceride and a reduction here in the counterplot. A small overlapping here, a prediction of 90% with this profile, highlighting by the scores, by IP, etc. that I don't show in here, that these are the change in this age, in the adolescence. What means? The only significant by volcanoplot is the monoacylglycerides. Monoacylglycerides has an important role in the production of pro-inflammatory signaling. Yeah? Okay. You must remember that this is information from the periphery. When we check 2030, there was a mirror of the information with respect to the children's. Here, the gangliosides, the glycolipids, the sphingolipids reduce at the serum. Some species, NAPS, NAPI, increase, with a small amount, very small amount, and 88% of the predictions. But what is this information? Is monoacylglycerides reducing, stronger reducing, because increase the species at 20, 30 years old, and highly related with the established pro-inflammatory signaling. In the older people, 31-40, other species was highlighted, more phospholipids, phosphatidylethanolamine, phosphatidylcholine, etc., and triacylglycerides, with 80% of the predictions, and all involving and saying, hey, we are involving this cell process in the endomembrane system are affecting, but severely. At 30 years old is severe this change. And we have a very high, or very strong reductions of these species at the serum. After 40 years, 41 years and older people, symptomatic, because all the previous range was asymptomatic. These are symptomatic people. Continue all these species, lipid species, but obviously associated with neurotoxicity, pro-inflammatory cytokines, phagocytosis, etc., and 93% of predictions, obviously in the symptomatic stage. Okay, we have to focus in the next two slides. The first, this, is what happened at the time. This is an idea of the Nelson Galvis, again, and word of the Laura Lozano. We have here is only a model. It's not the true, it's not the true no, because it's based in our data. It's true. We have confidence on that, but I mean respect to we must to increase, to expand the cases and to revise it thus with no 13 people, if not with 1,000 people, is the same behavior of the curve. But this model, advised by Dr. Daniel Camilo Aguirre, here we have no carriers and here carriers, and the more critical part is in the adolescents. The more important change is in the adolescents. This is only ApoE33. This is all people independent of the ApoE. And this is the inflection point between the children's, adolescents, and adult. Yeah, change completely this profile of lipids. And very interesting, Laura Lozano helped. This is very crucial support in our lab right now is doctoral students to standardize SIMOA, a single molecule array that we could to transfer to import to Colombia, our start country in Latin America with this kind of equipment to check the more advanced early biomarkers, protein biomarkers in biofluids. Mainly we study GFP, neurofilaments, phosphatau 231, phosphatau 217, and all of this, how is in the publication say that this is sensible, start from 24 years old. And that there is a special change in or stronger response in women. But the interesting is that the protein biofluid markers is detectable in in blood from the 20 years old. Sorry. From the 20 years old, that is the same point. Before or 20 years old is not possible by proteins in biofluids. And the second that I mentioned is the sex. We have here a heat map. This is a woman. Here's a man's heat map. It's a no carrier. No carrier is the 0 point. So black is white, and the red one is the change. Yeah, the fold change. Some of them were pivalos, significant pivalos. But here we have the 3,3 apoisoforms with changing in all the classes of lipids. And here is Aliria, that I know that all the people love the results of Aliria, that here, Aliria have a high, some of lipids high, and the rest of those are almost white. But when we check others, for example, carrying ApoE, one allele of ApoE4, the high life is increased very, very fold change. Okay, then the apoisoform, you know, the community know, the sex, heart, risk, susceptibility to increase the problems with the lipid metabolism related with the brain impairment. Nelson, in advice with Dr. A los dos los puse Nelson. El segundo es Daniel Camilo Aguirre. Is an epidemiologist. He suggests us that we were analyze blind, this is blind for genetic information, only based in the lipidome, which was the profile, the clustering that mentioned related with the clinical sociodemographic characterizations. This analysis rescued 5 profiles based in the lipidome with 5 different lipids involved in age profile, some of them talking about risk, some of them talking about protection, some of them relating more with aging, other with the younger people, and younger people, carriers, or depending on the apoisoform. Then we have a lot of work in the future to understand us. And those profiles in the adult people was related with the neuropsychological assessments mentioned or supporting the protection of the risk, alteration in the cognitive impairment. Okay. I'm going faster in this last part for mention that the people in the lab has working in the immunofluorescence, trying to check what happened with enzymes, with the proteins in the postmortem tissue. That's protein that is very important in the lipidome, related with myelin and neutral lipids. And also in collaboration with Kenneth Kosick, collaborating with the GNA, they in his lab prepare or analyze the single nuclear RNA, and we could understand partially how some genes are related with the lipid metabolisms, with the proteins involved in the lipid metabolisms. Why, partially, no? Different kind of ApoE change, in what kind of cells change, yeah? And what is the response of the cells response associated with that? Then the the final view is that understanding the emissions of lipids as biological biomarkers, but understanding the lipid metabolisms per kind of cells, the relation between them, the relation of the brain with the blood, the understanding the communication between between organelles, then we can detect these very early signals that could be the inspiration for the designs of the primary prevention and therapeutics. But the more important, confirm and support that the healthy lifestyle is the real prevention strategy, and why we predispose our organisms to develop this kind of diseases. Then with this study, we have a proof of concept. We could verify that exists an initial metabolic disbalance of the components of the neurovascular unit, impacting the homeostasis, releasing specific lipids to the periphery as early marker of cognitive impairment, but very, very early. Okay, we are working in collaboration with the Universidad de los Andes, some blocks from here, and develop design nanoparticles inspired in the proteins that regulate the lipid metabolisms, for tools, the diagnostic tools, very early, the predisposition. Obviously, this apply for the sporadic people. We have the machines working for the sensibility, the high sensibility. Thank you to the NIH. Until now, I have not found them to continue right now. And this is possible. Thank you to all the researchers, Dr. Lopera for the inspiration, David Aguillon for his completely confident and friendship, Stella, Ken, all the people working with us in a very confident manner. And this possible, definitely possible, thanks to the students that love the students. Thank you very much for supporting and to believe in this project. Thank you very much to you.
Thank you, Dr. Cardona, for that amazing presentation. I think that's very clear that having a healthy lifestyle is central to life. We have time for only one question.
Only one? Okay, 2 persons.
Doctora Cardona, muchas gracias por su presentación. Una pregunta, ¿cómo considera usted que, por ejemplo, estas tendencias en alimentación, por ejemplo, las dietas keto, pueden influenciar, probablemente o no, no sé, justamente esa es la pregunta, si usted considera que estas pueden influenciar de alguna manera el trayecto hacia enfermedades neurodegenerativas?
¿Dijiste la keto?
Sí, señora.
Ok, bien. Yo no soy nutricionista, entonces probablemente no tengo toda la visión, pero desde la... Ay, estoy respondiendo en español. Ok. Resulta que yo creo que todo depende de la oferta y la demanda, ¿cierto? Yo qué le doy a mi organismo, pero cómo lo consumo, ¿cierto? Y ese balance, ¿cómo lo mantengo en el tiempo? Si nosotros comemos 3 veces al día, los afortunados, pues tenemos que hacer muchas actividades para que definitivamente esa energía sea correctamente canalizada. Entonces, el punto está más ahí. El punto está en que si como muy bien, pues tengo que gastarme esa energía muscularmente, cognitivamente, emocionalmente, para que todo esté en balance. Ya si tú le pones, porque por ejemplo ahora está de moda la dieta ceto, ¿cierto? Pero tú dirías, no, es que le estás quitando toda la fuente energética y tu cuerpo, tu osteogénesis Bueno, el doctor ahorita Naya nos puede explicar mejor. Depende del colesterol, porque es que el colesterol produce células progenitoras, es la fuente de células progenitoras. Entonces, todo lo que es renovación depende de los lípidos. Entonces, no sé esas implicaciones a largo tiempo que vamos a tener, no lo sé, ¿cierto? Ya sabemos que hay una actriz muy famosa que ha sido muy estricta en su dieta keto y está muy enferma. Entonces, eso nos podría dar algún indicio, pero la clave está en lograr ese balance. Pero actividad cognitiva, emocional, física, o sea, no es solamente una. ¿Había otra pregunta de Laura? Ok, gracias.
Thank you again, Dr. Cardona. Up next, we'll have Dr. Wait a minute. Up next, we'll have Dr. Juan Manuel Anaya, who will discuss health and disease. What can we learn from the centenarians? Dr. Juan Manuel Anaya Cabrera is the leader of the Colombian Centenarian Alliance. He trained in rheumatology at the University of Paris and an immunologist with studies at the Pasteur Institute at the University of Texas at San Antonio. He holds a PhD in biology from the University of Antioquia. Centenarian Project aims to research different clinical genetics, epigenetics, and related biomarkers of longevity and resistance to chronic diseases in Colombian centenarian population. Please, Dr. Juan Manuel, the floor is yours.
Centenarios. Es correcto. Perfecto.
Perfecto. Aquí tiene el micrófono, puede pasar con los botones.
Paso con esta.
Exactamente.
Okay. Bueno, muy buenos días para todos. Extremadamente complacido de estar aquí con ustedes, aprendiendo de ustedes, viendo cómo esta área de la salud cerebral toma una fuerza cada vez mayor, apreciando mucho el entusiasmo y el compromiso del doctor Hernando Santamaría, a quien le extiendo mi más sincera gratitud y felicitación por este evento. También le agradezco mucho que me hayas permitido dar esta charla en español. Sin embargo, mis diapositivas estarán en inglés. Lo que yo les voy a contar es una historia que estamos construyendo con un grupo de amigos con el fin de aprender de longevidad saludable. Y esta presentación la hago precisamente en nombre del programa de longevidad saludable y de la Alianza Centenarios. No sin antes reiterar de nuevo mi gratitud a Hernando Santamaría por su amable invitación. Los centenarios, como ustedes lo saben, son las personas que han logrado llegar a la edad de 100 años o más. La longevidad es un problema de salud pública mundial y Colombia no es la excepción. Colombia, de hecho, es el país de las Américas que más rápido envejece. El índice de envejecimiento, que se calcula por el número de personas mayores de 65 años sobre el número de personas menores de 15 años por 100, es actualmente alrededor de 72, según cifras del DANE. Pero como ustedes lo ven en la diapositiva, para el 2050, este índice de envejecimiento será de 175.3. Luego, la rapidez con la que Colombia envejece es enorme. Las causas son múltiples. En primer lugar, y ustedes lo saben también, la tasa de natalidad en Colombia ha disminuido significativamente en los últimos 11 años. por encima del 35%. Las mujeres jóvenes no se quieren casar. Hoy en día en Colombia se permite la ligadura de trompas a partir de los 18 años por voluntad propia. Muchas mujeres jóvenes lo hacen y los hombres tampoco quieren hacerlo. Y la mayoría, o un número importante de ellos, buscan hacerse la vasectomía después de los 18 años. Luego la natalidad se estima cada vez va a ser menor. Por otro lado, la juventud en capacidad de producción está emigrando. Muchos jóvenes hacen parte de la diáspora colombiana. Pero al mismo tiempo, gracias a los servicios de salud, en este momento un poco deteriorados, pero de todas maneras, gracias a los servicios de salud, a la vacunación, a la atención primaria, etcétera, cada vez más hay más personas adultas. Luego, la longevidad es un problema de salud pública. El fenotipo extremo de la longevidad son los centenarios. Si nos situamos en una curva de Gauss, vamos a ver que en la mitad tenemos el promedio de longevidad y en los extremos ese 5% corresponde al fenotipo de los centenarios. Luego se convierten los centenarios en un fenotipo altamente informativo, que teóricamente, de acuerdo a las leyes de estadística, sería también muy homogéneo. Sin embargo, vamos a ver que no es así. De todas maneras, este grupo poblacional, como dije, es altamente informativo y nos va a permitir aprender no solamente de la longevidad saludable per se, sino muy probablemente, es nuestra estimación, nos va a permitir aprender de los factores protectores de las enfermedades crónicas que ellos no padecen, como lo vamos a ver a continuación. Finalmente, y no menos importante, los centenarios y la longevidad en general tienen un impacto importantísimo en la economía conocida como economía plateada, de lo que no mencionaremos el día de hoy. Los centenarios, a pesar de que sean relativamente frecuentes, su localización es muy difícil y hay una dicotomía enorme en Colombia y en el mundo entre las bases de datos de los centenarios y la localización de los mismos. En Colombia en particular es muy difícil encontrar a los centenarios por varias razones. En primer lugar, los centenarios mueren súbitamente, de un día para otro. Les voy a contar una experiencia. Cuando fuimos a Cali a ver centenarios, en la logística nos demoramos aproximadamente 2 meses, mientras hacíamos todos los ajustes para poderlos atender. En ese periodo de tiempo fallecieron 40 centenarios. Fallecen de un momento a otro. Las causas son diversas, por supuesto. Pero esta es una de las razones por las cuales encontrar a los centenarios es difícil. Más aún, esto tiene una repercusión importante desde el punto de vista demográfico, porque en Colombia, desafortunadamente, no es necesario presentar el certificado de defunción para las honras fúnebres. De tal manera que esos centenarios siguen, entre comillas, vivos en las bases de datos por periodos muy largos de tiempo. Se les sigue asignando la UPC y no solo eso, sino que siguen votando en las elecciones. Es algo muy particular a nuestro país, pero es lo que hay. Por otro lado, los centenarios migran muy frecuentemente. Los cuidadores de los centenarios resultan ser sus familiares en primer grado, sus hijos, sus nietos, etcétera. Y a los centenarios se les lleva de una ciudad a otra. Hemos visto centenarios de la costa que viven en Fusagasugá, por ejemplo, y están 3, 4 meses y luego se regresan a Soledad, como el caso de algunos centenarios que hemos visto. Lo cierto es que, a pesar de todo esto, los datos del DANE indican que en 1953 habían 529 centenarios en Colombia. En el 2023 se estimó que habían 19.400 y para el 2070 se estima que pueden haber alrededor de 45.000 centenarios. Quizás la cifra sea mayor, pero son los datos oficiales con los que contamos. El inicio de este proyecto tuvo lugar luego de un análisis retrospectivo de la base de datos de una EPS del régimen subsidiado tenía alrededor de 2.500.000 afiliados. Y para tener una aproximación inicial acerca del fenómeno de los centenarios, decidimos hacer un análisis retrospectivo de esa base de datos en la que comparamos los centenarios. Ustedes lo ven en la cuarta columna de la diapositiva. Comparamos esos centenarios, 2.964, con personas adultas mayores de 60 a 99 años y personas jóvenes de 18 a 59 años. ¿Y qué observamos? Lo primero que observamos es que la mayoría fueron mujeres. Ustedes saben que desde el punto de vista biológico, las mujeres viven más que los hombres. Sin embargo, en Colombia hay un fenómeno adicional para esta explicación y es que los hombres mueren tempranamente a causa de la violencia. En segundo lugar, la mayoría de los centenarios afiliados a esta EPS estaban en el área urbana. Encontramos que habían ciertas características desde el punto de vista clínico, como por ejemplo una tasa muy baja de diabetes. Asimismo, las enfermedades cardiovasculares y cerebrovasculares fueron muy bajas en esta base de datos. El cáncer fue extremadamente bajo. Asimismo, encontramos que la respuesta a la COVID-19 fue mucho mejor que la de los adultos mayores. Ustedes saben que uno de los factores de riesgo importantes de mortalidad en COVID-19 era ser adulto, sobre todo mayor de 70 años y obeso. Curiosamente, los centenarios no solamente tuvieron una tasa de infección muy baja, que puede ser atribuida al aislamiento, sino aquellos centenarios que sufrieron de COVID, la tasa de mortalidad fue significativamente menor a la de los adultos mayores. Luego, esta primera aproximación nos hizo entender que los centenarios son, sin lugar a dudas, una población aparte. En el camino de este estudio he aprendido que efectivamente los centenarios desarrollan muy poco enfermedades crónicas no transmisibles y tienen una muy buena respuesta a ciertas infecciones. Con este primer análisis decidimos entonces generar un proyecto para estudiar a los centenarios de Colombia, desde el punto de vista sociodemográfico, desde el punto de vista clínico y desde el punto de vista bioquímico en general, con el fin de hacer una aproximación translacional. Comparto contigo la opinión, hacer investigación translacional en Colombia es una utopía, pero los científicos perseguimos utopías, entonces hay que seguir haciéndolo. Tenemos todos los sesgos del mundo, no tenemos un solo peso, pero no sabemos hacer otra cosa. La hacemos a veces mal, pero es lo único que podemos y queremos hacer. De tal manera que a pesar de todas esas circunstancias, en este proyecto hemos concebido una metodología translacional que les voy a explicar a continuación para tratar no solamente de entender la longevidad saludable, sino apostarle a encontrar biomarcadores de protección de las enfermedades crónicas no transmisibles que ellos no sufren, que en definitiva son cáncer y enfermedades autoinmunes. Demencia lo veremos también a continuación. Algunos de los centenarios no son dementes, la gran mayoría lo son, pero algunos no lo son. De tal manera que ustedes ven en esta gráfica cuál es la metodología de estudio. Los centenarios son invitados a participar luego de la firma del consentimiento escrito por ellos o por su cuidador o familiar en primer grado, y nosotros les tomamos una serie de datos mediante un cuadernillo que hemos realizado, que tiene aproximadamente 305 variables semiestructuradas, algunas de pregunta abierta, otras de preguntas dummy, y esos datos antropométricos y clínicos los integramos en un repositorio. Al mismo tiempo, pedimos autorización para una toma de sangre, y esa toma de sangre la dividimos para química extendida, lo que he llamado así, como para un biorepositorio para estudio posterior de biomarcadores. Con todos esos datos, hacemos minería de datos nos ayudamos de inteligencia artificial para responder preguntas eventualmente pertinentes e importantes que sean útiles para la comprensión de la longevidad saludable. Las respuestas de estas preguntas no son otra cosa que conocimiento eventualmente nuevo que va a servir para los longevos que vienen atrás, lo que se queda en ese promedio de la curva de Gauss que nosotros no estamos estudiando. Ese cuadernillo de variables incluyen variables demográficas, clínicas, nutricionales, funcionales, cognitivas, de estilo de vida, prosociales, bioquímicas, inmunológicas, por supuesto, y en algunos casos estamos tratando de hacer imagenología cerebral. Igualmente, en algunos casos, cerca de 45 centenarios se han beneficiado de un estudio de capilaroscopia para evaluar el lecho ungueal capilar con el fin de tenerlo como proxy del estado vascular cerebral. Todavía no tenemos esos análisis hechos, pero estamos en curso. Ustedes ven aquí las principales herramientas o escalas que nosotros aplicamos para la evaluación de los centenarios. Y como se pueden dar cuenta, evaluamos la fragilidad, la sarcopenia, la percepción, la autopercepción de su salud, la calidad de vida mediante el cuestionario de la OMS. Vemos su estado nutricional, igualmente su independencia mediante el índice de Bartel. Igualmente estudiamos su capacidad funcional, su movilidad, evaluamos el riesgo de caída. Y tenemos 3 herramientas que, si bien no han sido validadas para centenarios, son las herramientas que hemos utilizado no habiendo otras. Todas estas herramientas que utilizamos sí han sido validadas, afortunadamente en español. Pero para evaluar el estado de cognitivo, utilizamos el Mini-Mental State Examination, que evalúa la alteración cognitiva. Empleamos el MOCA, que ustedes conocen muy bien, para hacer un screening de alteración cognitiva, y utilizamos el Clinical Dementia Rating, que nos sirve para evaluar la severidad de demencia. Igualmente evaluamos la depresión, la ansiedad, los estilos de vida, el insomnio y la severidad del mismo, el bienestar y variables prosociales. Al mismo tiempo, como les dije, tenemos una serie de preguntas sobre el estado de salud, antecedentes, etcétera, etcétera. ¿Cómo se lleva a cabo este proyecto y cuál es la parte eventualmente traslacional que queremos aplicar al mismo? Ustedes lo ven en la parte derecha de la diapositiva. Luego de ese consentimiento informado, tomamos una cantidad de datos, incluyendo datos biológicos. Tenemos un biorepositorio. En ese biorepositorio nos entretenemos para ver cómo podemos evaluar los biomarcadores de interés, ya sean proteómica, metabolómica, genómica, etcétera. Hacemos un estudio de esos resultados con análisis bioinformático para tener correlaciones clínico-biológicas y finalmente poder, en la medida de lo posible para nuestra población, este es un estudio que hasta ahora no se ha realizado en nuestro país, de tal manera que vamos a poder entender cómo se comportan estos factores que estamos estudiando en nuestra población. Y con esos factores vamos a tener información acerca de cuáles son los factores, a su vez, de riesgo o de protección. Posteriormente vamos a poder entender cuáles son los factores de longevidad saludable para que puedan ser aplicados de manera preventiva y eventualmente en una cuarta fase poder hacer personalización siempre y cuando exista la participación activa de la comunidad, en este caso de los longevos. Ese es el sistema 5P de medicina translacional que hemos preconizado desde tiempo atrás en la época en que trabajábamos en autoinmunidad. Y eso es lo que estamos haciendo actualmente. ¿Qué resultados quisiera compartir con ustedes el día de hoy de manera muy breve? Quiero contarles que como este es un estudio en curso y es un estudio al que le he dedicado los últimos 3 años y estoy muy interesado en aprender precisamente de longevidad saludable, pues hemos tenido que comenzar desde el principio, a pesar de que hayan trabajos en centenario en otros países, pero nuestra población nos ha enseñado que no todo el comportamiento de la salud es similar a la de otras partes del mundo. Por lo tanto, nos dio curiosidad de saber cómo era el comportamiento de la calidad de vida y el bienestar en los centenarios, cómo se comportaba el aspecto nutricional de los centenarios. A medida que hemos estado viendo los centenarios, hemos visto que no todos son iguales y que esa homogeneidad esperada desde el punto de vista estadístico por tratarse de un fenotipo extremo en realidad no existe. Entonces nos dimos a la tarea de aproximar una nueva taxonomía de los centenarios y finalmente, y lo más importante para mí fue o ha sido poder estudiar el comportamiento autoinmune e inflamatorio en los centenarios. Comencemos entonces por qué hemos aprendido de la calidad de vida y el bienestar de los centenarios. Ustedes ven los resultados en promedio en esta tabla de las herramientas que utilizamos y pueden darse cuenta que el promedio de calidad de vida fue 49.7 utilizando la escala de la OMS y desde el punto de vista de su bienestar, el promedio fue 26.95. Son promedios que son menores a lo que se ha reportado en centenarios en otras partes del mundo. El resto de resultados lo ven ustedes en la tabla. Lo interesante es saber cómo se correlacionan estos resultados entre ellos. Y ustedes ven aquí una tabla de correlación en donde pueden observar cómo la calidad de vida se correlaciona de manera significativa con el bienestar, lo que es una obviedad, pero también con el desempeño físico, el estado cognitivo, el estado nutricional y la independencia funcional. De manera muy interesante, ven ustedes que hay una correlación inversa altamente significativa entre la calidad de vida y la depresión. Con respecto al bienestar, algo similar ocurre con la depresión. Hay una correlación inversa importante y muy significativa y, por supuesto, hay una correlación entre el bienestar y el estado cognitivo medido por el MOCA y, por supuesto, una correlación con la calidad de vida. Estos resultados varían ligeramente en función del sexo. Entiéndase, en las mujeres no es idéntico como en los hombres, sabiendo que en nuestra población la calidad de vida de los centenarios hombres es muy superior desde el punto de vista estadístico a la calidad de vida de las mujeres, por diversas razones. En primer lugar, la violencia, sobre todo. Ustedes pueden darse cuenta que la calidad de vida en las mujeres se correlacionó con el desempeño físico, con el estado cognitivo, con el estado nutricional y con la independencia funcional. Y a su vez hubo una correlación inversa con la depresión. Con respecto al bienestar sucedió algo similar. ¿Qué tan bien o mal nutridos están nuestros centenarios? Y esto es un aspecto muy importante porque he aprendido hasta ahora, gracias a lo que muchos de ustedes me han enseñado, que el trípode sobre el cual se sostiene la longevidad saludable es el sueño, el bien comer y el ejercicio. De tal manera que la nutrición para nosotros era un aspecto importante y por eso evaluamos su estado nutricional en esta población. Ustedes ven los resultados demográficos en esta tabla, ven que la mayoría de nuestros centenarios para este análisis fueron mujeres, la mayoría vivían en área urbana. Tengo que insistir en que la mayoría de nuestros centenarios son de muy bajo nivel socioeconómico y la mayoría son iletrados. Aunque el estrato socioeconómico en los centenarios en Colombia deba ser redefinido de acuerdo a las escalas que utiliza el DANE. Hemos visto, por ejemplo, centenarios en Sucre que viven en una casa de bareque, tienen una estera para dormir, tienen una letrina fuera de su casa y tienen una cocina de leña afuera y al frente de su casa un potrero con 500 cabezas de ganado y jamás fueron al colegio. No les importa absolutamente nada y viven mejor que muchos de nosotros eventualmente. Eso para hablar un poquito de la riqueza en los centenarios. Vean ustedes cómo la mayoría de los centenarios eran edéntulos y el 50% usaba prótesis dental. La fragilidad se observó en el 75%, la sarcopenia en el 73%. A pesar de eso, la depresión, que es algo tan importante en estos centenarios, fue observada solo en el 15%. La ansiedad en el 1%, alteración cognitiva en el 73%, insomnio en el 19% y la satisfacción con la vida en el 86%, que es una paradoja para mí muy interesante que tiene que ver con la resiliencia de este grupo poblacional. A pesar de todas las dificultades y las adversidades que han tenido durante su vida, cuando se les pregunta cómo se sienten, si querían volver a vivir lo mismo, etcétera, responden muy favorablemente. Desde el punto de vista nutricional, el 36% estaban malnutridos y con riesgo de malnutrición el 47%. Luego, la gran mayoría sufría de mala nutrición o desnutrición. ¿Qué observamos en cuanto a las correlaciones con las medidas antropométricas y las otras variables clínicas de las que les he comentado hasta ahora? Pues el estado nutricional en los centenarios se asoció con un estado funcional y cognitivo. Tiene el estado nutricional un impacto muy importante y significativo sobre la salud mental y funcional, sobre el desempeño físico y, por supuesto, ese estado edéntulo de los centenarios es característico de la desnutrición que ellos padecen. De hecho, las tres principales características o problemas de salud que tienen los centenarios son la hipoacusia, la presbicia, por diversas razones, maculopatía del adulto, cataratas, etcétera, y el ser edéntulo. Y eso tiene un círculo vicioso que me enseñó el profesor Lopera, asociado directamente con la demencia, sobre todo aquellos que tienen presbiacusia, que es un problema de salud pública al que muy poco se le presta atención. Desde el punto de vista de la clasificación de los centenarios, pues no todos los centenarios son iguales y hay una clasificación clásica que seguramente ustedes conocen. Aquí traigo una copia del título de este artículo de Jessica Ebert de hace más de 10 años, que clasificó a los centenarios en sobrevivientes, en retrasadores y aquellos que se escapan, todo referido al estado de salud. Los sobrevivientes son aquellos que tienen enfermedades crónicas no transmisibles que han adquirido antes de los 80 años y llegan a ser centenarios. Los que retrasan o los delayers son aquellos que tienen enfermedades crónicas no transmisibles después de los 80 años y llegan a ser centenarios. Y los que escapan son aquellos que llegan a los 100 años sin enfermedades crónicas no transmisibles. Y la mayoría de los centenarios publicados o las series de centenarias publicadas en el mundo se refieren a que estos centenarios, la mayoría, han escapado, o sea que llegan a la edad de los 100 años sin enfermedad crónica no transmisible. Eso es lo que dicen los artículos y la literatura. Sin embargo, en la experiencia hemos observado que ese paso de los 99 a 100 años hace desde el punto de vista biológico que muchas de estas enfermedades que ellos pueden traer se estabilicen. Tal es el caso de la diabetes, de la hipertensión y de la enfermedad renal crónica. Por esta razón, y dada la experiencia que estábamos adquiriendo, decidimos aproximar una nueva clasificación de centenarios utilizando la enorme clinimetría que hemos empleado en el estudio de esta población. Mediante un análisis de conglomerados utilizando componentes principales, encontramos 3 conglomerados o clusters que ustedes ven en la figura. El primero de ellos, en color azul, corresponde a lo que hemos llamado centenarios vulnerables. Son centenarios que llegan muy mal a los 100 años, llegan dementes, la mayoría están postrados, no necesariamente tienen enfermedades crónicas no transmisibles, pero están en condiciones físicas prácticamente de invalidez. Esos son los vulnerables. En la parte media ven un grupo en amarillo que hemos llamado resilientes porque efectivamente llegan a los 100 años con algunas características de deterioro de salud, pero en términos generales están bien. Y hay una población que es la altamente informativa, que ustedes ven en gris, que corresponde a lo que ellos hemos llamado vigorosos o antifrágiles. Esa población, que corresponde al 15% de los centenarios, de acuerdo a este estudio, llega en muy buenas condiciones de salud, como lo vamos a ver a continuación. Aquí están algunas de las variables que distinguen estas tres poblaciones. Les pido el favor de que fijen su atención en la columna de vigorosos para ver la relación que hay con los otros dos grupos. En primer lugar, fueron mucho menos las mujeres. Eso quiere decir que mucho más hombres llegan a los 100 años en estado vigoroso. Desde el punto de vista de medidas antropométricas, los centenarios vigorosos tienen mejor fuerza de prensión y una mayor circunferencia de pantorrilla. Asimismo, su estado nutricional, su independencia, su capacidad funcional y el desempeño físico es mucho mayor que los otros dos grupos de centenarios. De manera muy interesante, no observamos en este análisis, no observamos que el grupo de vigorosos tuviera demencia o un estado cognitivo severo. Luego, este es el grupo al que hay que prestarle toda la atención. Ahora les voy a mostrar cómo tampoco tienen cáncer ni enfermedades autoinmunes. Como era de esperarse, entonces, la calidad de vida en el grupo de los vigorosos es mucho mayor que en los otros dos grupos. Algo que me llamó mucho la atención cuando hicimos este análisis es que desde el punto de vista del estado inflamatorio, ustedes saben que hay un concepto emitido por el grupo italiano del doctor Caruso hace muchos años atrás, que se conoce como inflammaging, y es un estado inflamatorio de la adultez y, ¿por qué no?, en los centenarios. Pues desde el punto de vista macro, si me permiten decirlo, cuando evaluamos los niveles de ferritina, de proteína C reactiva y de velocidad de sedimentación globular, no encontramos que hubiera diferencia en estos grupos, y eso nos llamó mucho la atención. Quisimos ir un poco más allá, no me voy a detener en los detalles porque todo esto son resultados eventualmente negativos, pero pudimos estudiar un panel de cerca de 20 citoquinas, tanto inflamatorias como antiinflamatorias, y ustedes pueden ver que desde el punto de vista estadístico encontramos una variabilidad muy grande, como era de esperarse, pero no encontramos diferencias significativas en cuanto a interleuquina 1 beta, 2, 4, 5, 6, 7, IL-6, etcétera, etcétera, interleuquina 10, por ejemplo, como tampoco encontramos en otras que han sido ampliamente estudiadas en la longevidad, por ejemplo, interferón gamma o interferón alfa. No encontramos esa diferencia en los centenarios que estudiamos. Ahora bien, es ampliamente conocido que el sistema inmunológico no es el mismo en la niñez, en la juventud, en la edad adulta y en los centenarios. No me voy a detener en esto, pero voy a traer a colación este resumen que muestra cuáles son las principales diferencias del sistema inmunológico en los centenarios. Los centenarios tienen una mayor cantidad de células T, tienen células TCD8 activas, tienen una disminución en el número de células B y aumento de las células que producen IgM, eventualmente como anticuerpos naturales. Desde el punto de vista de las células asesinas naturales, NK cells, tienen un aumento en su capacidad de producción de citoquinas y de citotoxicidad, con un aumento en la producción de interferón gamma en los centenarios. Desde el punto de vista de actividad de los neutrófilos, hay un cambio en la quimiotaxis y en la adherencia observada en los centenarios, y desde el punto de vista de inflamación, los centenarios tienden a tener un perfil antiinflamatorio y no proinflamatorio, como eventualmente se podría considerar. Y finalmente, la parte inferior de la diapositiva es muy ilustrativa porque si bien los adultos están propensos a todas las enfermedades llamadas de la edad, los centenarios o las retrasan o simplemente no las padecen. En resumen, los centenarios tienen una mayor actividad antiinflamatoria, mayor número de células TCD4, tienen mayor número de células diferenciadas CD8, así como un mayor número de células B vírgenes, y su actividad y función de células naturales asesinas está preservada. Finalmente, les voy a contar qué hemos observado con respecto a la autoinmunidad en los centenarios. Lo primero que hicimos, luego de atenderlos y tener un cuestionario previamente validado que habíamos hecho hace unos años atrás de enfermedades autoinmunes y de antecedentes de autoinmunidad en la familia, hicimos un screening de un número importante de autoanticuerpos. Y nos llevamos la sorpresa que ustedes están viendo en esta tabla. Y es que un número importante, cerca del 44% de los centenarios, tenía autoanticuerpos. En particular, anticuerpos antitiroideos, anti-TPO, antitiroglobulina. Pero también tenían anticuerpos como el factor reumatoide, anticuerpos antifosfolipídicos y otros. Luego, lo primero que hicimos fue mirar qué se había reportado en la literatura al respecto, y efectivamente hay reportes de centenarios eventualmente sanos que presentan fenómenos de autoinmunidad, que en este caso no se puede considerar autoinmunidad latente, entiéndase por autoinmunidad latente la que precede la autoinmunidad clínica, porque a los centenarios no les da enfermedades autoinmunes. Sin embargo, esa autoinmunidad, cualquiera que sea, muy probablemente autoinmunidad natural, esa autoinmunidad en los centenarios colombianos fue significativamente mayor a la que se ha reportado en 10 estudios previos que revisamos de la literatura. Ahora bien, cuando fuimos a ver qué enfermedades autoinmunes tenían estos centenarios, efectivamente encontramos que 8 de los 114 centenarios que examinamos tenían hipotiroidismo, con una prevalencia que es baja del 7%. El hipotiroidismo suele ser mucho mayor, sobre todo en las personas adultas. En los centenarios fue la única enfermedad autoinmune que pudimos comprobar. No observamos ninguna otra enfermedad autoinmune llamada órgano específica, ni ninguna enfermedad autoinmune sistémica. Fíjense ustedes que hay algunos centenarios que tienen anticuerpos altamente específicos de artritis reumatoide, anticuerpos antipéptidos cíclicos citrulinados, anti-CCP en la tabla, pero ninguno de los centenarios que vimos y examinamos tenía artritis reumatoide. Un número pequeñísimo tenía anticuerpos anti-RO. No observamos síndrome de Sjögren en los centenarios, ni tampoco observamos vasculitis, ni observamos lupus eritematoso sistémico u otras enfermedades autoinmunes clásicamente conocidas. Luego pudimos comprobar efectivamente esa paradoja de la autoinmunidad en los centenarios. ¿Por qué llamamos una paradoja de la autoinmunidad? Porque cuando se hacen las revisiones de la literatura y aún entre colegas nos preguntábamos qué íbamos a observar, pues mucha gente, lo mismo sucedía en oncología, esperaba que en los centenarios se observara enfermedades autoinmunes, como también se observara cáncer. Sin embargo, ni lo uno ni lo otro se observan en los centenarios. ¿Cuáles son las hipótesis para explicar este fenómeno? Pues bueno, el inflammaging, en realidad que se menciona tanto, en los centenarios no existe. Desde el punto de vista genético, los centenarios no son portadores de alelos de riesgo conocidos de autoinmunidad. Los alelos del HLA, DRB1 y DQ son ausentes. Los alelos de autoinmunidad conocidos son ausentes en los centenarios. Y asimismo, otros polimorfismos asociados con autoinmunidad, como TNF-alfa, PTPN22, etcétera, etcétera. La inmunosenescencia es otro fenómeno que no se observa claramente en los centenarios y que lo hace la hace una población diferente. Es probable que ellos no hayan tenido la hiperestimulación necesaria para hacer enfermedades autoinmunes en la adultez mayor. haya un fenómeno de proteodinámica que sea distinto al del adulto mayor. De esta manera que esas son las hipótesis que esperamos trabajar en un futuro para comprender mejor la observación que hemos hecho, como otras observaciones que hemos hecho y que hacen parte de la fenomenología de estudios inicialmente descriptivos que posteriormente convertiremos en analíticos. Para terminar, entonces, quiero recordarles que Colombia enfrenta una transformación en su pirámide poblacional, que es el país de las Américas que más rápido envejece, que los centenarios se convierten en una población tremendamente atractiva para el estudio de la longevidad saludable y que no todos los centenarios son iguales. Hay centenarios que son vulnerables, otros que son resilientes y otros que son vigorosos. He mencionado ya varias veces esa ausencia de enfermedades crónicas no transmisibles, en particular cáncer y enfermedades autoinmunes, como también la buena respuesta a ciertas infecciones, lo que hace que el concepto de inmunosenescencia deba ser por lo menos reevaluado en esta población. Y todas estas preguntas que nos hacemos son interesantes porque detrás de esa pregunta hay una razón o deben haber algunas razones biológicas que nos deben incitar a hacer estudios un poco más profundos al respecto para entender esa fenomenología. Sin lugar a dudas, yo aprendo que la actividad física y un buen estado nutricional son fundamentales para la longevidad saludable, y a los jóvenes les comparto que la investigación en los centenarios ofrece una maravillosa oportunidad para entender no solamente la longevidad saludable, sino para el descubrimiento de los factores de protección de las enfermedades que ellos no padecen. Los médicos y en la escuela de medicina siempre somos llamados o incitados a pensar en los factores de riesgo. ¿Qué hace que a una persona le dé una enfermedad? Pero el Homo sapiens, como todos nosotros aquí, estamos hoy en este salón gracias no a los factores de riesgo, sino a los factores de protección, como sucede en los centenarios. Luego hay una avenida inmensa por estudiar y yo quiero contarles que desde ese punto de vista están todos ustedes invitados a ser parte de este proyecto. Nosotros hemos querido que este proyecto sea lo que se conoce como un proyecto de ciencia abierta, es decir, que la comunidad científica sepa eventualmente en tiempo real qué es lo que estamos haciendo. Y para eso creamos esta página, cuya dirección es muy sencilla, www.coolsen.org, en donde ustedes van a encontrar todo lo que estamos haciendo y sobre todo lo que queremos hacer. Vamos a ver si realmente somos capaces de hacerlo. Ahora, este trabajo no se pudiera haber realizado en este corto periodo de tiempo sin la colaboración y participación generosísima de ciertas instituciones como la Universidad de la Costa, Biotech la Clínica de la Costa en Barranquilla, donde incluimos un número muy importante de centenarios, el CREA de la Universidad del Rosario, Metacor de la Universidad de los Andes y Autoimmunity Lab aquí en Bogotá, como de investigadores asociados que han sido fundamentales en el desarrollo del estudio. Quiero mencionar particularmente a dos de tus estudiantes, Hernando, la doctora Sandra Castelblanco y Alejandra Malpica, que entre otras han sido mis profesoras de geriatría ad honorem. A ustedes, muchísimas gracias por su atención.
Doctora Naya, muchísimas gracias por su presentación. Ojalá que el proyecto de alianza salga muy bien y podamos todos eventualmente ser centenarios. Lastimosamente no tenemos espacio para preguntas, pero debido a que tenemos un pequeño momento de break, tenemos solamente 5 minutos disponibles para poder hacer break. Entonces, si tienen preguntas para el doctor, por favor acérquense, aprovechenla, háganlas. En 5 minutos retomamos con la siguiente presentación. Afuera hay una estación de café y hay un pequeño refrigerio para ustedes.
Tienes que avisar que hubo un pequeño cambio de plan, entonces no se va a hacer el diálogo, sino que va a haber una receta para almorzar. ¿Listo?
Welcome back. We would like to inform that we are having small changes in our agenda. So today we had this discussion panel at the lunchtime. Now we're going to have a free lunchtime, so you're going to be able to go and have your lunch in your own time and come back to at 1 p.m. Now we will resume the presentation with Dr. Catherine Rankin presenting social emotional brain networks in aging and neurodegeneration. Dr. Rankin is a professor in the Department of Neurology at the University of California, San Francisco, UCSF, where she specializes in the neuropsychological, neuroanatomical, and genetic basis of social-emotional behavior in aging and neurodegenerative diseases. She holds a PhD in clinical psychology and a master's in theology from the Fuller School of Psychology and studied psychology at Yale University. Her research uses neuroimaging to investigate empathy, theory of mind, and the processing of social cues. She also leads biostatistics and bioinformatics efforts within collaborative research networks and contributes to UCSF Precision Medicine Knowledge Network. Please welcome Dr. Rankin with an applause.
Thank you so much. I am so pleased to be here, and I just want to say thank you to the folks that have been such wonderful hosts to me, Nano and Diana and Pablo. You guys have been amazing, and I'm really happy to be able to be here and speak with you guys about this topic. As you heard, I'm a neuropsychologist. I work in research and do clinical care also with patients at University of California, San Francisco. And I have found, as I've traveled around the world, everywhere I go, I find that really we don't get taught this in medical school, in graduate school. Psychiatry programs, neurology programs, they really don't teach how the brain does social and emotional functioning. So I'm really happy to have a chance to talk about that. And I'm going to spend some time today really just teaching about how I view these neural circuits and how they function. So, sure. So I don't have any particular disclosures, just federal and private funding in the US. I'm going to change this. So today I'm going to particularly look at the neural circuits that seem to be most important for supporting social and emotional functioning in the brain. I'm going to talk about what those networks are and what they do, and then I'm going to talk a little bit more about how they interact, because of course social functioning is complicated and these networks interact, but I'll try to make it as straightforward as I can. And I want to make sure I have my timer going here. There we go. All right. So we're going to start with individual networks. I want to say first, for those of you who are not used to working with neurodegenerative disease, this is a point that's very important that we in my field think about all the time. About more than 15 years ago, we realized that there is intrinsic architecture in the healthy brain that is particularly attacked by neurodegeneration. And let me be more clear about that. So we all have intrinsically connected networks in our brain. These are functional networks. Often the structures in the brain are anatomically distant from one another, but those structures, those networks of structures, function together. Those structures are wired together into intrinsic networks. And each of those networks has a particular function. So there's a network for finding things. If you're looking for your friend in a crowd, how you do that visual search. There's a network that supports that. How you put together grammar and language. There's a different brain network that supports that. Well, about 15 years ago in neurodegeneration, we realized, actually my colleague Bill Seeley realized, that when he looked at maps of brain atrophy in patients with neurodegeneration, the particular focal atrophy that went with different neurodegenerative disease syndromes matched the networks that are happening normally. So those intrinsic networks, they're focally damaged in neurodegeneration. So, for instance, with Alzheimer's disease, the earliest network to be hit is the memory network in the brain. Whereas with non-fluent aphasia, a disorder where grammar and motor speech is affected, the network that is affected is, in healthy people, a motor speech network. And so this made me very interested because I care about social functioning in the brain. And I realized I work with patients called behavioral variant frontotemporal dementia patients all the time. Now, these individuals, even very early in the disease, have a lot of damage to parts of their brain that we associate with social and emotional functioning. Those are the earliest things to be hit. We all know later on in the disease, there are much more broad deficits in executive function, but early on, it's the insula, the cingulate, the other parts like the orbitofrontal cortex. And so their primary initial symptoms in this syndrome are highly disrupted social behavior. So that made me think, what's going on here with the intrinsic networks? And there is a particular network in healthy people that is initially damaged in BVFTD. So that at that time, I was like, oh, so damage to that network is enough to create that BVFTD syndrome. Why is that? What exactly is that network doing? Now, that network is called the salience network. And it turns out that it's important for paying attention to, alerting to social and emotional cues. Now, this network originally it developed, it evolved so that we would pay attention to important things in our world. We all have many, many stimuli coming into our brains all the time. I could be looking at the color of the seats. I could be looking at the pictures on the wall. I could be hearing the air coming through. I could be hearing rustling. I could be thinking about my internal signals. Maybe I'm hungry. Maybe I'm trying to remember to call somebody later on this evening. All kinds of stimuli come in. But only certain stimuli are really important, right? Rather than the seats, your faces are more important, right? Knowing that, say, if I were a cave person and I'm sitting in a clearing weaving a basket, I'm very focused on that basket. I want that to work properly. But if a tiger jumps into the clearing, suddenly the basket is not important. I have to pay attention to the tiger. This network helps us shift our attention to what is most important to us. And by important, I mean what is important for our survival, what is important for our well-being, our health, what is important for our thriving. And so you might wonder, why does this network have so much to do with social and emotional functioning? Well, it turns out because we are social animals, what other people are thinking or feeling are sometimes critically important signals for our health and our thriving and our survival. If we do not pay attention to, if we don't know how to read other people, like say I'm rude to my boss regularly, I might get fired and I don't have a job and I don't get to I don't get to support my family. If I am on a date and I don't pick up on signals and I don't know how to behave, then maybe I don't get another date with that person and maybe I fail to have children. So for the organism, for humans, we need to be aware. If somebody rolls their eyes at us, that's an important signal. We need to immediately tune into it and notice what that means and start thinking about it. So that's what the salience network does. It helps us pay attention to those social signals. And we have to do that to maintain adequate social status and support in our community. So what happens when the salience network, everybody's different. Everybody has a salience network in their brains, but some people's are very tightly connected so that they're very, very in tune and strong. And some people's are very weakly connected, right? So it turns out that when people's networks are tightly connected, it actually looks like anxiety. You might see that they're alerting to lots of signals as being very socially important. Maybe some that aren't that important still seem very important. Whereas the question when we first discovered this network about 15 years ago, it looked like these behavioral variant patients, their salience network was being damaged. We thought, oh, it's probably poor social skills. But my group really set out because we wanted to prove that. And a lot of the work I'm going to talk to you about today is the work we've done in the last decade to show that this network really does directly support social functioning. So I want to give you a picture of how I think about these networks. Obviously, we perceive many things from our internal and external environments, and we need to decide what's important. So that's what this core social network in the middle, that's the salience network. And we used to think of, of the salience network in a way that it was alerting to and focusing attention on what was important. Since then, we've realized it's a little more specific than that. The ventral part of the salience network is about noticing that stimuli, picking it out, noticing it. Whereas there's another network, more dorsal, that is involved with holding on to and paying attention to that network. So one thing to know about this network is it's not just cortical, it's also including much of the autonomic nervous system and subcortical structures. So one of the things you heard from Agustin yesterday was that interoception is very important when we're trying to measure this disruption to the circuit. That's a good way to get at it. So parasympathetic functioning. Now, I'm not going to talk about that very much today because I don't do psychophysiological experiments, but a lot of my colleagues around the world have done so. Virginia Sturm, Fiona Comfer, obviously Augustin, have done wonderful work with this. So I want to point that out. But really, like I'm saying, in order for somebody to have this BVFTD syndrome, they can actually, in a subset of patients, have only damage to those subcortical circuits, and their cortex might be preserved. I did a paper with a wonderful, she was a postdoc at the time in my group, and now she's faculty member, just a wonderful person to work with, Kamalini Ranasinga. And I did a paper showing that a third of the patients in our sample really just had subcortical damage, but their behavior was just like the ones with the cortical damage. So I want to make a point here. I think that in my field, there are two aspects of work that we do. One of them is really called affective neuroscience, right? We want to understand how the brain works. We want to understand the mechanisms of how things change with disease. And we might create tests just for our study. We have a question, we create a measure, and then we use it for that study. We don't ever think anybody's going to use that measure again. We get our answer, we publish the paper, we're done. But there's another aspect that I also am very committed to, and it's really socio-emotional neuropsychology. How do we measure these as a community? What tests are we going to use to get at functions, and are we going to be able to use them with individual patients? Ideally, these measures can be used with individuals. They can be used by people repeatedly, and we have normative data about it. And for me, knowing what's normal versus abnormal is important, so you can understand what this test score meant for your patient. But also, it's very important to know that these tests can predict what brain circuit is being affected. And so I'm going to mostly talk about our measures that do that. So one of these measures that we've done neuroscientific work on that is also a neuropsychological measure is a very quick, easy little measure called the Revised Self-Monitoring Scale. And it's measuring just one simple aspect of empathy. It's measuring our ability to pick up on subtle social cues in a room, like with the person. So if you're with somebody and they make a joke and you find it rude, you find it kind of inappropriate, but you're being polite, you'll laugh and, oh, that's great, yeah. But the other person, if they're not sensitive to your cue, they will think, oh, she thought that was funny. If they're sensitive to your cueing, they will say, ooh, I think she thought that was rude. They'll notice it and they'll adjust their behavior. So this measure is something, it's just 13 items, and we ask caregivers and informants of other people who interact with the patient to answer, is the patient good at this or is the patient bad at this? So just 13 items. And obviously it's been translated into Spanish if you're interested in using it. But let me show you the work we've done with this. So first of all, it does correspond beautifully with gray matter volume in this salience network that I'm talking about. It's also very good at distinguishing patients with a behavioral presentation like BVFTD and some of the other ones from others who don't have a behavioral presentation like Alzheimer's, and also very different from healthy controls. It is actually very sensitive to the early signs of BVFTD, and it is sensitive all the way along as BVFTD patients get worse. So it's good with progression. And it also, the brain changes correspond with progression in the salience network. So this is a lot of different evidence that we've shown over the years that this measure is related to salience network structure and behavior, but we still have the question, does it correspond with connectivity, function in the brain in that salience network. And it turns out when we did the studies of this, first of all, we saw that across healthy people and patients, there was a lot of variability in salience network function and tight connectivity versus loose, not so good connectivity. And it turns out that the score on this measure directly correlated with that. So there was a lot of variability, and we could actually predict the score based on connectivity in a cortical network and or a subcortical network that was centered around the right anterior insula. So this is, again, the brain was being able to predict the behavior, how another person views you, and it was accurate. But this is one thing that I found very important about this. When we looked just in healthy people, no patients, in younger and older adults, we saw that RSMS score predicts the strength of their salience network connectivity on brain scans. So this measure really does reflect what's happening functionally in the brain. Now we also, all these studies were really about whether structures were tightly or loosely connected, but we also wanted to do some work about whether those structures were communicating directionally with each other. So we did something called dynamic causal modeling of this resting state functional data. And we were able to show, sorry, frog in my throat. I think I'm okay. I'll just speak with the frog for a minute. So we found that when we looked at how the structures within the salience network communicated with each other, we found some very important things, even in healthy people. We found that particularly a structure in the salience network called the periaqueductal gray. Now that's a structure you might have heard of before, and it has to do with fight, flight, freeze. It's a very basic in the brainstem sort of structure. And what we found was that when the PAG spoke to the rest of the brain, when direction of communication was out to the cortex, people were more empathic. Everyone said, that person picks up on my signals well. They're right about how I'm feeling. But when the cortex was talking back to the PAG and suppressing or changing those signals, people were rated as not as empathic. So the PAG communication with the rest of the brain was important. And FTD patients also really did not show good PAG function. Their brain was not receiving the PAG signals. So I want to talk about a second network. Oh, thank you so much. I want to talk about a second network that is important to us. And in the same study, one of the things that was seen was that there was a syndrome called semantic dementia, which we now call semantic variant primary progressive aphasia, that also had a correspondence. So there's a network there that we healthy people used all the time, and no one had really looked carefully at what its healthy function was. And so the patients that have damage here, they have damage in the temporal lobes, particularly the anterior temporal lobes. They have damage to the caudate, the medial part of the orbitofrontal cortex. That seems to be the primary damage. And in our patients, we have a tendency, they start either on the left side of the brain or on the right side of the brain, but eventually this syndrome, both sides of the temporal lobes are damaged. So what we see here is a network that happens in healthy people, and it really does two things. It pulls together our knowledge about the world, our learned knowledge about the world, and how we feel about that information. So basically, if I say, do you like ice cream? Or do you know what ice cream is? Most of you have experienced ice cream, and so you have knowledge. You could describe, well, it's cold and it comes in different flavors and it has this texture. You've experienced it. That's your knowledge. That comes from the temporal poles. But when I ask you, do you like strawberry ice cream better than chocolate ice cream? You're going to have an opinion about that. And it's going to be different across this room. And that opinion is based on your experience, your personal experience. You have a valuation of strawberry ice cream. And whenever I say ice cream and strawberry ice cream, you immediately link what it is with how you feel about it. The brain is programmed to do that. We have a superhighway to connect what things are and how we feel about it, who people are and how we feel about them, places, activities. We automatically put what it is with how we feel. And the how we feel part of it is the subgenual cingulate, and it's the ventral striatum, the caudate, and the nucleus accumbens in the brain. And this network is called the semantic appraisal networks because it's looking at the semantics of something and the appraisal of it. Now, why is this important for social and emotional functioning? Now, I really appreciate Matt Lambon-Ralph and others who have done a lot of work looking at what this network does for all of our understanding of the world, all of our semantic understanding of the world. And in particular, Richard Binney and Richard Ramsey looked at the fact that when we are trying to understand a concept, the temporal pole is a hub for information, but that temporal pole is taking information from lots of different regions. So if I say dog to you, and you need to think about what a dog is, you might think about how that dog sounds. So you're going to activate a spoke that is an auditory spoke. You might activate a sensory spoke, how the dog feels when you pet it, or how it feels when you pick it up. How big is it? So that's proprioception, that's texture. You're also thinking about what they look like. So these are different spokes that are all contributing to one core social concept, or one concept. But social functioning is no different. When I ask about what a meaning of a social concept is, if I say that person is arrogant or prideful, you kind of know what that is because you have experienced it and you can picture it. But there are different spokes in the brain that allow you to understand social concepts that you might not need when you think about a dog or a chair. So valence is very important. So in terms of core socio-emotional networks, in addition to the one I just said, salience, semantic appraisal can fit in this schematic. It's really, salience, attention, and knowing what things are and how we feel about them really play together. They go together. And we have a few tests of this sort of thing. And again, this is, we have a Spanish version. We have many different language versions of this test. This is one we made to really show whether our patients understood social semantics. It's a quick little test, 16 items. They're easy, medium, and difficult. And we're really just looking at, we give them a word and we give them 4 pictures, and they have to pick the picture that goes best with the word to show that they understand the concept. And it turns out that our behavioral patients have a lot more trouble with this, which is good news. That's what we were hoping to see. But more importantly, when we look at the structural anatomy, we really see that those orbitofrontal structures, the caudate and the orbitofrontal regions, that's the valuation part of the brain, is contributing to function on this. And if it were just a language test, just is that a chair, is that a bus, those parts of the brain probably wouldn't be, we know they are not as active. Whereas when we look at a social concept, that orbitofrontal valuing needs to be there. It gives us precision about our understanding of those social concepts. So another thing that's important about what this network does is it actually helps us read emotions. And when you think about it, that actually makes sense because there's been a lot of research that shows that when you're reading an emotion, you actually embody the emotion a little bit, particularly if the other person's signal is kind of ambiguous or hard to read. Like if I showed you a video with somebody making an emotional, expressing themselves, and I turned the sound off, you might lean in, and I asked you, like, what emotion are they showing? You might lean in, you might start doing, kind of not even realizing, doing that emotion on your face. That's really typical. We humans do that. And when we're blocked from doing that, like if researchers have somebody hold a pencil in their mouth so they can't do the face motions, or when people get Botox deadening of their facial expression, they actually are poor at reading emotions, and they actually are shown to have less amygdala process. Being able to pick up your interoceptive signals is part of the information that lets you know what's happening. And so when we give patients a test of emotions, we are asking them to activate this network. And it turns out this is really the network that corresponds with emotion reading. So we did a test. Again, we were trying to make tests that were easy and quick for our patients. So this is the DART, the Dynamic Affect Recognition Test. It's just 12 items, very quick little, 3 to 5 minutes to administer. And importantly, we have videos of individuals expressing emotions, but the script, the thing they're saying, is neutral. We had the actors use the exact same words and express anger and surprise and fear because the scripts were so neutral, it didn't matter. But all of their body language, their face, their voice, their body language is very clearly and consistently expressing the emotion. And then so somebody watches the video and we ask them, what emotion did you just see? And the nice thing about this is I want to highlight my colleague, Nahuel McGrath-Guymet, who is at FLENI in Buenos Aires. He has now filmed a Spanish version of this task. And we have, because it's in Argentina, I now realize Argentinian Spanish is not understood by people in other countries. And so he did 2 versions. He did an Argentinian version that they're validating there. And he did another version that was like neutral Spanish that should be more comprehensible. So let me actually play a video. You can see one of the items.
¿Viste esto? Mira, mira la página 13. Parecen iguales, ¿verdad? Esto cambia las cosas para nosotros, ¿no es cierto?
So you can see that she is, oops, sorry, didn't mean to replay it. You can see that she is surprised. So, and it's just another quick little test. Now, I want to put a pitch out. We're trying to work with some folks here, and we also are interested in working with individuals who also have healthy controls, because we're trying to get enough healthy controls so that we have a normative basis to administer this task to patients. So if you want to collaborate on that, we would love to give you a copy of this. So importantly, this does correspond. Emotion labeling ability does correspond with the salience network. Now, I'm first showing you structure here. When we look at the structural regions of the brain that it corresponds with, you can see it's mostly temporal lobe. The blue is well, let me put it this way. What we did with this was we subtracted out all the non-social cognition, and the red is really just the social cognition parts of the DART, and it's right-sided. It's got the head of the caudate. We have some orbitofrontal regions. So we also did a connectivity study on this, and we showed that this was actually on a slightly different test, but same, another test called the EET, Emotion Evaluation Test. Same principle. It's videos of people expressing an emotion. And it turns out that the semantic appraisal network focused around the right anterior temporal pole and those orbitofrontal regions are directly correlated with people's actual performance on the test. So we also saw that this test is very good at picking up deficits in our patients that have already a behavioral deficit in social cognition, particularly those who have that right temporal BVFTD sort of flavor. So it's very good at that. And another thing we found is that we tried to give these dynamic tests with the videos, and we gave just static face pictures to patients, which a lot of folks do when they test emotion. But it turns out that you really need the video-based test to pick up those nuances. It's hard for patients to do those static picture-based tests. And I have to say, like, when we were trying to distinguish a right temporal variant from a frontal variant, we need the dynamic tests. Now, I want to say just one quick point, because I was highlighting like Julia's work. Julia Ulagood I'm so lucky to be working with her. She's a fellow in my group. And she's worked with Jolande Pinjenberg at Amsterdam for the last couple of years, putting together the International Right Temporal Variant Working Group. And I just want to make a quick point about why this is relevant for social cognition testing. So we as a group, my group in particular, has made a lot of social function tests, but I feel very uncomfortable going to other countries and other cultures and saying, hey, just take my test and translate it, or just adapt my test. Because I just believe that in individual countries, in individual cultures, it's going to be very, very different. So I love the model of what Julia and Yolanda now I'm doing in this group is we've actually pulled together, not because we have a big research grant, but because we have interest, pulled together more than 100 researchers at more than 30 centers, more than 30 centers, but in 30 countries. And we all meet regularly and talk about how to diagnose these conditions and what the symptoms are. And we talk about what the gaps are in social cognition tests. And so we decide, here's a symptom, like for instance, being able to measure rigidity, mental rigidity and preoccupations in our patients. Here's a symptom we need to measure. Let's define it carefully together in agreement. And then everyone does their own test of that. And we might learn from each other or make tests that are similar to each other or harmonize them. But the point is the concept, and we all help each other to get the right kind of testing. So I really encourage that. As if you're interested in test development, that's a wonderful way to do things more internationally, starting not with a Western, United States or European test and then adapting it, but starting with the concept and having everyone build what's right for their country and their culture. So I'm going to spend the last few minutes talking about complex interactions among these networks. And so in my rubric here, you can see the core two socio-emotional networks I've talked about. But when we look at the right side, look at planning and control, we actually have a few different functions that influence our social cognition. One of them is executive functioning, and one of them is memory. It's not core, but it's important. And let me give you some examples of why. We did a study that was a task-based fMRI study of moral reasoning. We did it with healthy older adults and with patients with BVFTD. And when we did this, we gave people moral dilemmas in the scanner. And when I talk about moral dilemmas, I talk about personal versus impersonal dilemmas. So let me make it clear what that is. The dilemma might be, okay, you have a little tiny boat and you are out in the water and a big ocean liner has gone down or a big ferry has gone down and there are lots of people drowning in the water. And there's one person over there and there are 4 people in the water over there. Which way do you go? Do you get the one person out of the water or do you get the 4? You can't get both. And when I ask that, most of the time people just say 1 versus 4. I'm going to do the math. 4 is more than 1. I'm going to go get those 4 people. And they don't really feel too bad about the fact that the 1 got left. They're just like, I'm making the right choice. I know that's the right choice. Get 4 people. But what if I told you the 1 person in the water was your nephew? Suddenly that changes the equation. Suddenly you're like, I can't just do the math anymore. You start to think about your nephew, your relationship with your nephew. You think about playing, maybe playing catch with him when he was younger, or how your sister is going to feel if you let her son drown. What will your relationship be like in the future? How much you love him. You think about all those things before you make your choice. So it turns out that when we are doing an impersonal dilemma, 1 versus 4, 1 stranger versus 4 stranger, we use the executive networks in the brain. We just do the math. But when we're doing something personal, when it becomes real to us, then we actually need to use our memory networks. We start to activate our schemes, our scripts. We think about the history and the future of that choice. But when we look at FTD patients, they don't do this. They tend to choose, even if it's their nephew in the water, they choose the 4 people, right? They make utilitarian choices, and they're not activating their default mode network as much. So we did a causality analysis, and the bottom line with this was, in healthy people, the salience network is a gating mechanism to choose whether the executive or the memory networks are functioning. And then BVFTD patients, that's dysfunctional. Let me just explain that a little bit better. So I'm gonna make sure my clock is working. So with a functional salience network, if I give you an impersonal moral dilemma, you're just gonna do the math. You're just gonna do executive functioning to figure it out. But if you realize, hey, that's my nephew in the water, then you're going to activate the default mode. And the salience network is what decides for you whether it's important and personal or not. However, with FTD, The salience network can't do that. It can't decide if it's important or personal. And so it doesn't know an impersonal from a personal dilemma, and pretty much usually activates the executive network as if it's doing math. And it turns out that this mechanism that we saw in frontotemporal dementia patients is the same mechanism that appears in autism. At the same time I was publishing this, I ran into Udin Menon at Stanford, and he had found the same thing in those autism patients. So this is the known gating mechanism of the salience network. We found the same thing looking at other kinds of higher social cognition. So when we think about theory of mind, reasoning, being able to infer what another person is thinking. We show videos to patients and they have to sort of decode, what did that person really mean to accomplish there? What were they trying to do when they said that thing? And it turns out that the salience network, when it's dysfunctional, the other parts of the brain don't really matter. If you don't have a salience network, you can't really use the memory and executive very effectively to decode what other people mean. And this is hot off the presses, literally just very, very recently. This is another fellow I have the privilege to work with, Sherry Chen, does amazing work with the cerebellum that I can't present today. But she did this study with functional connectivity among these networks and showed that not only, well, let me just say, in controls, it turns out that one network, the default mode, that memory network, is really important. It needs to be receiving information from the salience network and from executive network. But in FTD patients, it's not. The default mode network, the memory network, is just talking to itself the whole time. It's not receiving information anymore in those patients' brains. And they are doing these theory of mind tests very differently. So I just want to summarize and give you guys a chance to ask questions, but I'm just going to repeat that these intrinsically connected networks are very helpful for us to understand social cognition, whether or not we work with neurodegeneration. It's very important for me as a dementia researcher, but it can be important for you in any work you do in psychiatry and neurology, with healthy people, with patients with psychiatric disorders. The salience network, again, that's helping pay attention to and alert to important cues. And we use things like the RSMS and other questionnaires. Also, interoception tests. If you can do that, that's very helpful. I know those don't really work in a real life setting, but if you can, that picks up salience network function. Semantic appraisal network is mediating emotional evaluations. So any kind of knowledge about social norms, social rules, knowing what expectations are, that's semantic appraisal network. Being able to pick up and decode what another person's emotion is and having categories for that, that's semantic appraisal network. So we use the SIVT so as an example, social interaction vocabulary test. We use dynamic emotion tests. And again, those dynamic tests, that's important. If you're going to test emotion functioning, it should be realistic. You know, we don't read each other's emotions by looking at a picture as much as we read each other's emotions by feeling what's happening in the room. And so we need to give patients the chance to show us that they can do that. And again, these networks work together with additional memory and executive networks during complex social cognition and moral reasoning. So I hope I've given you a general overview to understand how these networks work for social cognition, and that I hope that you will be able to sort of think of it this way and use this when you're trying to understand what's happening with your patients, and maybe you can see how their behavior shows you a little bit about what's changed in their brain or what's happening in their brain. And I want to just acknowledge, I'm so thankful to our patients and their families, and I'm so thankful to all the people that I work with. I have so many collaborators, I can't even fit them on the slide. I have so many wonderful people that have passed through the Rankin Lab, fellows, postdocs, graduate students, and a lot of them have contributed directly to this work, so I'm just really grateful. Thank you very much. I think we have time for questions, if there are any.
Yeah, thank you very much, Doctor. It was an amazing presentation. We have a couple questions here, so I'm going to go first, then here.
Hi, thank you for your presentation. I really enjoyed the talk. I had a question regarding the lack of insight from the patients, because I think clinically it's very relevant, for example, behind the repetitive behaviors and all. Have you evaluated its relationship with the networks, the lack of insight and the networks?
That's a great question. So I think everybody heard it, but how does lack of insight get reflected in these networks? And it turns out we have done a number of papers on insight and self-awareness. And one of the things that we found was that one of the things I didn't say this very clearly, but the semantic appraisal network contains some reward structures. The nucleus accumbens is in there, which is part of reward responsiveness. And when we look at insight, self-awareness, it turns out that when you don't have good reward circuitry functioning, you don't have insight. It actually, when you think about what insight is, it takes cognitive energy to pay attention to yourself and be judging yourself and evaluating whether or not you're doing what other people are expecting you to do. And when we actually find it rewarding to be right about that, we find it rewarding to be accurate. And that's an adaptation that humans have had, that we do like being right about the world, and we want precision with our perceptions. And so what we found was that patients that had subcortical even damage to their reward pathways were worse at being aware of their own deficits. We also found that being insensitive to punishment cues. So again, punishment so the reward circuits are part of the semantic appraisal network. Punishment cues are more salience. They're more lateral orbitofrontal, and so that's more salience network. So when we don't really have any care about what other people are going to think of us, if we don't care whether we're right or wrong, or whether our behavior is good or bad, then we stop having insight. And so we just stop being able to pick up on those cues that we've made an error. And so that's what we see. And so it's a little bit of both networks, but they're contributing different functions. Great question.
First of all, thank you so much for your wonderful talk. I have many questions, but I'm just gonna make one, and hopefully maybe we can talk later.
Absolutely.
Because I'm very interested in this topic. So my question is regarding, you mentioned at the beginning that people with better reading of social cues also had anxiety, but I'm guessing this is a normal anxiety.
Mm-hmm.
But that made me think what happens in people with anxiety disorders. And also, in the same spirit, what happens in people with other neuropsychiatric conditions that are bad at reading stimuli, such as depression and schizophrenia.
Mm-hmm. Great question.
What happens in the networks that you show in this context?
Yes, thank you. I thank you for the opportunity to clarify that, actually, because I cut it a little short for your talk, and I'm glad I get to talk about it. So really thinking about when I said that the salience network, high levels of connectivity in individual has been shown to correlate with higher anxiety and low levels poor social function. One of the things I didn't say was that there's actually a U-shaped curve to the amount of salience network connectivity. Our happy spot is sort of in the middle somewhere. That's where we're most sensitive to social cues, but we're also not over personalizing it. So when your salience network is too tight, because that network is supposed to be picking up on threats, really, things that are potential rewards, but also potential punishments in the environment that are relevant to us. That's where that high anxiety comes from. And when you look at people with, for instance, post-traumatic stress disorder or generalized anxiety disorder, high levels of connectivity in the salience network is part of their phenotype. We've found that in the brain. And so therapeutically, when we treat them, we want to see that salience network connectivity relax a little bit. What we really want is we want more interaction with other networks in the brain. So the salience network is noticing, but then passing on the information to other networks that can then do the work. And so that is very important for other disorders. One of the things that we see in patients, I do a good amount of work looking at the difference between our behavioral variant FTD patients and our dementia patients who just lose salience network function altogether and have bad behavior, versus patients who have psychiatric disorders. And what we see is that patients with psychiatric disorders, depression, anxiety, they actually often have higher salience network functioning. And they are actually usually very aware of social cues. People with depression, studies have suggested that they're better at reading emotions than people that are not depressed. They're more attuned. And a lot of people with anxiety, it's mixed. Sometimes your anxiety makes you look away from the face and you don't pick up others because you're just too anxious about it. Other times you tune in and you're focused. So different anxiety phenotypes have different degrees to which they're sensitive to social phenomenon. But overall, they're much more aware of you than somebody with a BVFTD. So that's a great question. I think I got all of it. I hope I remembered everything you asked. Okay. Is there time for more, or is that? No more time. Okay, thank you so much.
Up next, we will have Dr. Brian Lawler. Give me a second to connect him. Dr. Lawler, can you hear us?
And yes.
We can hear you as well. Excuse me for all the waiting. We are ready for you.
Okay, great.
Give me a moment, I present to you. We can see your screen as well. Perfect. As I said before, now we have Dr. Brian Lawler, who will address why we need to take a brain health approach for people living with dementia. Dr. Brian Lawler is professor of old age psychiatry at Trinity College Dublin and a founding director of the Global Brain Health Institute at Trinity. He is geriatric psychiatrist with clinical and research expertise in dementia, late life depression, loneliness, and brain health. His work spans over 30 years in the development of care services for people with dementia, with a research focus on early detection, prevention, and new treatments. Dr. Lawler, the floor is yours.
Many thanks, and it's great to be here with you, even though it's virtual. I'd love to be there in Bogotá. And I'm going to talk to you about why we need to take a brain health approach to dementia. And what I want to talk about is really why, if we take this approach to dementia, we can improve the quality of life of people living with dementia. So here's an outline of my talk. First of all, I'm going to give you a case study of a person with dementia. Then I'm going to talk about brain health, what it is and how we can achieve it, even if you have dementia. Then I'll talk about modifiable risk factors for brain health in dementia, particularly polypharmacy, delirium, vascular risk, and lifestyle factors. I'll then move to tell you about a trial I coordinated a number of years ago called NILVAD in mild to moderate Alzheimer's disease. And this was very much a natural experiment where we can explore the impact of risk factors on brain health in dementia. I'll then return to the case study and emphasize why taking a brain health approach to dementia can stabilize trajectories and improve quality of life for people living with dementia. So first of all, the case history. This was a 72-year-old man, a former bus driver with a 3-year history of Alzheimer's disease. He had a background history of vascular risk factors, hypertension, diabetes, and also had both of his hips replaced. He had some depression and anxiety and some sleep problems. He was living at home with his wife, and his wife had severe arthritis. She was quite incapacitated, and his son would come on a daily basis to help out and take care of the shopping. And over the last number of months, his son had noted that his father had become quite withdrawn. He was staying in the house, and he'd had 2 falls. And on examination, his walk was quite slowed down. He appeared a little bit drowsy. And when his blood pressure was checked, his blood pressure sitting down was 130/90, but when he stood up, it dropped significantly, particularly the systolic blood pressure. His fasting blood sugar was high, suggesting that his diabetes was poorly controlled. And his cognitive assessment at the time was 16 out of 30, which would indicate a moderate degree of dementia. Although, he was only 3 years into his Alzheimer's disease, and up until a few months ago, he was more functioning in the mild range. So this was a bit of a surprise that he was more moderately impaired at this stage. He was on a number of medications. He was on a thiazide, a diuretic for his blood pressure. He was on a calcium channel blocker for his blood pressure. He's also been prescribed an antipsychotic by his general practitioner, supposedly for anxiety. He was on an SSRI for depression. He was on an antidiabetic medication, metformin. He was also being prescribed a diazepam for sleep and anxiety. And he was on an alpha blocker for a problem with his prostate. So I'll return to the case history in a little while, and it'll make a lot of sense, I think. So first of all, what do we mean by brain health? Well, brain health is the optimum state of cognitive, emotional, psychological, and behavioral functioning. So it's not the absence of a brain disease. It's the best brain fitness that you can achieve. Achieving brain health is about protecting your brain. It's about reducing risk to your brain and building your brain's capital. You do this by promoting the brain's plasticity and improving connections. And essentially, achieving brain health is getting the most out of your brain over the life course. So everybody can achieve brain health. Brain health is for everybody. And you can have brain health even if you have dementia. So brain health is about, first of all, reducing risk to your brain, looking at risk factors such as traumatic brain injury, smoking, hypertension, cholesterol, diabetes, depression, loneliness, hearing loss, and visual loss, and addressing these risk factors to reduce the risk to your brain. It's also about promoting protective factors. So protective factors includes physical exercise, education, social connection, cognitive stimulation, education again, and arts and creativity. But I want to emphasize that brain health can be achieved even if you have dementia. So how do you achieve brain health if you have dementia? Well, essentially, you identify, reduce, and promote. You identify the risk factors to the brain health, even if you have dementia. You reduce these risk factors. You look at risk reduction strategies, even if you have dementia, for the person with dementia. And just think back in the case history, there are a number of different risk factors to that Mr. A's brain health, and there are a number of ways that you can reduce the risk to his brain. And then you promote protective factors. So what are the risks to brain health in people living with dementia? So there are a number of areas that are really important. First of all, drugs, medications that people are taking. Many older people, and particularly people with dementia, are taking lots of different medications. They may have polypharmacy, or some of the medications are inappropriately prescribed. And this poses a risk to the person with dementia, to their brain health. Another risk to brain health in people living with dementia is delirium. This is the development of an acute confusional state, usually related to infection. And when people develop infection in the context of dementia and develop delirium, that can speed up the rate of progression. People with dementia can also have lifestyle factors that can impact on their brain health, just as it could impact on our brain health of an individual who doesn't have dementia. So excess alcohol, low exercise levels, isolation, loneliness, all of these can impact our brain health, but also can impact the brain health of a person living with dementia. And very importantly, vascular risk factors like high blood pressure, hypertension, but also low blood pressure and blood pressure variability. These are all risk factors for brain health for everybody, but they're also risk factors for the brain health in people living with dementia. So let me tell you a little bit more about polypharmacy and anticholinergic burden from drugs that people could be taking, particularly older people and people living with dementia. So polypharmacy is defined as taking 5 or more drugs. And if you're taking 5 or more drugs, you're more likely to fall, you're more likely to have adverse events and unscheduled hospitalizations. And we know that for older people, if you're on more than 5 medications over a period of time, that's associated with increased risk of developing dementia. We also know that inappropriately prescribed medications, medications that can combine and interact with one another, can have cognitive impairing effects. A very good example of potentially inappropriate medication prescribing is antipsychotics for a prolonged period of time. And we know that antipsychotics are associated with accelerated cognitive decline. And you probably remember that Mr. A was on quetiapine, an antipsychotic. Also, drugs that block the cholinergic system in the brain and have high anticholinergic burden are associated with increased risk of developing mild cognitive impairment and cognitive decline over time, particularly if you are ApoE4 positive. And we know that older people who are taking drugs that have high anticholinergic burden over a period of time are at increased risk of developing mild cognitive impairment and Alzheimer's disease when they're followed over 9-10 years. Now, what about delirium? Well, we know that delirium is common in people with dementia. Up to 50% of people with dementia may develop a delirium, and the prevalence range is very wide, from 22 to 89%. But when you develop an episode of delirium in the context of dementia, this accelerates your rate of progression. So, for example, if somebody with a dementia develops an episode of delirium, there's an additional almost 2 points increase in progression on the MMSE per episode. So that's probably about at least 6 months of accelerated progression. The more episodes of delirium you have, the greater the decline. But the important point about delirium, it's a risk factor for brain health in people living with dementia, but it's potentially preventable. Now I want to turn to blood pressure behavior and dementia risk. So it's well known that hypertension increases your risk of developing dementia, but also prolonged hypotension or low blood pressure over a long period of time can increase your risk of developing dementia. What's less commonly known is that orthostatic hypotension or low blood pressure when you stand up is also a risk factor for the development of dementia, as is blood pressure variability. So blood pressure variability is where when your blood pressure is measured at different visits over a period of weeks or months, if it's variable, this is a risk factor for the development of dementia. So blood pressure variability are associated with dementia risk. So I want to tell you a little bit more about orthostatic blood pressure behavior. So blood pressure changes, systolic blood pressure changes of greater than 20 when you stand from a sitting position, or greater than 10 millimeters of mercury when you stand from a sitting position in diastolic blood pressure. This is what we mean by orthostatic hypotension. And so when people have these blood pressure shifts when they stand up, like Mr. A, they can get orthostatic intolerance such as lightheadedness, dizziness, but also they can fall. And when you take blood pressure using a sphygmomanometer, you can define orthostatic hypotension hemodynamically as I have above here. However, early orthostatic hypotension can also be related to systolic blood pressure recovery changes at 30 to 60 seconds. So, and you're not going to pick this up using a standard sphygmomanometer. You have to use other methods as shown here this is a FiniPress, which is looking at arterial blood pressure in the finger. And this is much better able to define early orthostatic hypotension. So this figure shows you the orthostatic blood pressure changes that can be picked up using this beat-to-beat measurement of blood pressure and heart rate using FiniPress or arterial finger blood pressure. And you see this delayed recovery of blood pressure at 30 seconds. And this can only be picked up using these arterial finger blood pressure measurements. You're not going to pick this early orthostatic change up on a standard sphygmomanometer. So when we looked at this orthostatic blood pressure behavior change, this early delay in blood pressure recovery, we looked at it in a group of people with mild cognitive impairment and compared to controls. And what you see here is that compared to the controls, people with mild cognitive impairment have this delay. But they have those people with mild cognitive impairment who actually went on to develop dementia had greater slower blood pressure recovery at 30 seconds. So this orthostatic blood pressure recovery change, this delay in blood pressure recovery following a standard that you pick up on the FiniPress measure seemed to predicted progression from MCI to dementia. So again, looking at survival estimates, people with MCI who had greater or slower recovery in their orthostatic blood pressure were more likely to progress to dementia. So in this trial, this nilotinib trial in Alzheimer's disease, this was a natural experiment where we could look at a number of these different risks to brain health in people with mild to moderate Alzheimer's disease and see what was the impact of, let's say, delirium, blood pressure recovery, blood pressure variability, and polypharmacy on the rate of progression and on adverse events in people with mild to moderate Alzheimer's disease. So nilotinib was a clinical trial with a repurposed calcium channel blocker, blood pressure medication. And it was a phase 3 controlled trial over 18 months. We randomized 511 people to either drug or placebo with mild to moderate Alzheimer's disease and followed them for 18 months. Unfortunately, nilotinib itself had no effect on disease progression. It was ineffective as a disease modifier. However, we were able to look at other risk factors on disease progression, as I mentioned, medications, delirium, and blood pressure behavior to see whether that impacted on the rate of progression or had any other adverse effects. And in this trial, we were able to measure medication use very, very finely and also looked at adverse events. So we had a detailed medication history from the patient, but also from caregivers. We were able to document all adverse events and serious adverse events, unscheduled hospitalizations, and unscheduled GP visits, including delirium. We had a lot of information about the drugs and their anticholinergic burden. And we were also able to define whether people were getting inappropriate medications. And this was defined using very standardized criteria. We also detailed measures of blood pressure behavior. We had blood pressure sitting and standing every visit over 18 months, which allowed us to look at blood pressure variability, which I've mentioned is a risk factor for dementia. And we were able to include blood pressure variability if people had over 3 office visits, and that would have been the majority of people that were randomized. And then on a subgroup, we were able to look at day-to-day blood pressure variability and blood pressure recovery using the home blood pressure measurements, this finger arterial blood pressure measurement in a sample of 55 people. So what do we find in terms of risks of brain health in these people with mild to moderate Alzheimer's disease? Well, we found that about almost 30% of people were prescribed an anticholinergic medication, which blocks the cholinergic system and is not good for the brain health of people with Alzheimer's disease, whose cholinergic system is already failing. We also found that those people with Alzheimer's disease who had a high anticholinergic burden, they seem to progress more rapidly on the functional scale, not on the cognitive scale. And this was more likely if they were ApoE4 positive, that if you were ApoE4 positive status predicted greater progression on both the CDR, which is a measure of disease stage, and also the functional scale, the DAD. So anticholinergic burden seemed to have an impact on progression. And what about long-term antipsychotics? About 12% of people were on long-term antipsychotics, and not surprisingly, this accelerated decline at 12 and 18 months. And again, particularly true for people who are ApoE4 positive. So long-term antipsychotics being prescribed to people with mild to moderate Alzheimer's disease accelerated their rate of progression. Now, what about potentially inappropriate medications? Over half the people with Alzheimer's disease were prescribed a potentially inappropriate medication, but almost a third were prescribed 2 or more potentially inappropriate medications. And the most commonly prescribed potentially inappropriate medications were benzodiazepines and SSRIs. And these medications were associated with more adverse events, more serious adverse events like deaths or hospitalizations, unscheduled hospitalizations, and unscheduled GP visits, but didn't have an effect on disease progression. So more adverse events, more hospitalizations, but not on progression. Benzodiazepines were commonly prescribed and one of the most common potentially inappropriate medication. 14% were on benzodiazepines. Benzodiazepines were not associated with greater cognitive decline, but they were associated with much more adverse events, delirium, and falls. We also looked at episodes of delirium. And what was interesting here was that people who had episodes of delirium during the 18 months trial, this accelerated their progression. And it accelerated their progression about 3 points on the ADAS-Cog over the 18-month period. And that's about a 3 to 6 months additional decline. So this would mean that if you could prevent one episode of delirium in these people with mild to moderate Alzheimer's disease, that would give the person an extra 3 to 6 months of quality of life. Something that's very much worthwhile. Now, what about orthostatic blood pressure recovery and blood pressure variability and progression? So looking at orthostatic blood pressure recovery, you could see that those that had slower blood pressure recovery tended to be faster progressors. And that was both for systolic and diastolic blood pressure. Also, blood pressure variability was associated with a more rapid progression. And those people with mild to moderate Alzheimer's disease who had the highest blood pressure variability, they seem to have more rapid progression. So what are the implications for blood pressure variability and orthostatic blood pressure behavior for brain health in people with dementia? Well, blood pressure behavior is abnormal in people with mild to moderate Alzheimer's disease. Blood pressure behavior like orthostatic hypotension, blood pressure variability can affect cerebral perfusion, and it could be important in modifying the trajectory of cognitive decline in people with dementia. One thing we have to be careful about is with blood pressure management, blood pressure treatment in dementia is not to be too aggressive and not to cause too low a blood pressure. We also know that polypharmacy can contribute to blood pressure behavior, and it needs to be studied also as a potential modifiable risk factor for progression in people with dementia. One of the things we know about continuity of care is that if people with mild to moderate Alzheimer's disease are receiving better and more continuous care from their family practitioner, they seem to do better. There's been a couple of studies in this regard showing that higher continuity of care in dementia is linked to safer prescribing, less polypharmacy, less potentially inappropriate medication prescribing, and also lower rates of adverse events, including delirium. The converse is also true, that lower continuity of care is associated with higher rates of hospitalization, more visits to the ER, more testing, and higher healthcare spending. So if you have more continuous care, better care, you're more likely to not to be prescribed inappropriate medications, and your outcomes are more likely to be better. So what does all of this mean? Well, in my view, if we take a brain health approach to people living with dementia, protecting their brains, reducing risk to their brains, even though they have dementia, we're more likely to be able to stabilize trajectories and improve their outcomes. In the NILVAD trial, we showed that if you are prescribed potentially inappropriate medications, you're more likely to have adverse events. If you have higher anticholinergic burden, you're more likely to progress more rapidly. If you're prescribed antipsychotics, you're more likely to progress more rapidly. If you have an episode of delirium, you're more likely to progress more rapidly. So if we take a brain health approach to people living with dementia and reduce these types of risks, we may be able to stabilize their trajectory. So treating and preventing delirium, decreasing adverse events, avoiding medications that can adversely affect cognition, addressing vascular risk factors, hypertension, orthostatic hypotension, atrial fibrillation, and now I would include orthostatic blood pressure behavior and blood pressure variability. All of these things can potentially protect the brains of people living with dementia and stabilize their trajectories. Obviously, we also need to adopt healthy lifestyle factors, because this may also be helpful in terms of stabilizing the trajectory in people with dementia. So if we go back to the case study, Mr. A, what should be done there in terms of risk reduction? Well, the risks that Mr. A was experiencing were the polypharmacy. He was on many different medications. He was on potentially inappropriate medications, benzodiazepines, antipsychotics. He was suffering from significant postural hypertension. He had a number of falls. He's also depressed and becoming isolated and socially lonely. So identify those risks and then reduce those risks. First of all, through deprescribing, looking at improving his diabetes control, activate him, connect him, and increase his level of support. So this is taking a brain health approach, trying to improve the brain health of somebody living with dementia. So let's see what happens if you reduce these risks once they're identified. So here's the follow-up on Mr. A. So what was done, his metformin was increased to get better diabetes control. The alpha blocker, which was causing postural hypotension, was stopped. His antipsychotic was stopped because he didn't need it. His diazepam was stopped because he was drowsy and it's an inappropriate prescribing. His citalopram was increased for his depression. So 4 weeks later, he's reassessed. His MMSE had increased from 16 to 20, which is a very significant increase, bringing him back into the mild range of dementia. His blood pressure, sitting and standing now, didn't show a significant postural drop. He wasn't falling anymore. His mood was better. He was less drowsy. And he was socially connected now, attending a retirement club. So you can see what happens if you reduce risks, protect the brain, address these risks to the brain. Even in somebody living with dementia, you can improve their quality of life, improve their cognitive score, and gives them back 6 months to a year in terms of a quality of life. So taking a brain health approach to people living with dementia, is this a new way of doing things? Well, if you rethink dementia from the perspective of brain health, it's a very positive thing. It changes the narrative around dementia. It's a positive value to achieve for somebody living with dementia. It brings hope. It gives them agency and pathways. There are things you can do. As I've emphasized about reducing risk to the brain, protecting the brain, focusing on what can be done, reducing risk, deprescribing, improving the medical care, monitoring and protecting the brain. So in a sense, the message for primary care is that we should be doing more, not less, to protect the brains of people living with dementia. There are challenges, of course, because this involves collaboration, coordination across care systems. It involves developing an integrated chronic disease care model. It does mean spending more time and more effort, working across different professional silos, working across medicine, social care, arts, voluntary agencies, and it involves leadership. So if you really want to change hearts and minds for brain health, it does involve leadership. Everybody wants change, but it's hard to change themselves. And again, leading change can be tricky, can be difficult, as this cartoon shows. But essentially, that's what we're trying to do at the Global Brain Health Institute, is to change hearts and minds for brain health. And as I said before, brain health is a human right. Brain health is for everybody. And it's not just for people who are healthy or people who are at risk. It's also for people who actually have a dementia. And I hope I've been able to show you that and convince you that if you take a brain health approach to people living with dementia, there's a lot we can do to protect their brains, to reduce their risks, and actually to help stabilize their trajectories and improve their quality of life and functioning. And I think I'll leave it there. Thank you very much.
Thank you very much, Dr. Lawler. I would like to know if someone has questions in here. We have just space for one question. So I would like to know if someone has a question in here.
I have one here. Someone sent me, delirium is a condition affecting older adults, which, if identified, requires prompt diagnosis. What are the most effective strategies for its detection in both hospital and community settings?
So could you repeat the question again for me?
Delirium is a condition affecting older adults, which, if identified, requires prompt diagnosis. What are the most effective strategies for its detection in both hospital and community settings?
So it's a really good question, and I was involved in a program that actually introduced a care pathway for detection of cognitive impairment, not dementia, but also cognitive impairment and delirium in a hospital setting. So first and foremost, it's around education and awareness. There's lack of understanding really of what delirium is and what it isn't. So education and training is really important. And also the introduction of screening tools, simple screening tools that can be used in the emergency department and right across the hospital. And having protocols in terms of, once you make once you screen positive, that there is an action. And I think one of the most effective tools and easy to administer in this regard is the 4AT. It's a very good screening tool for delirium. It's one that we introduced in the acute hospital setting where I worked. And we embedded that then into a care pathway, and it has significantly increased the diagnosis of delirium and improved management.
Thank you, Dr. Lawler. I think that's all for our part. Thank you so much for your presentation. Thank you so much. Please bring it again for Dr. Lawler.
Thank you.
Up next, as Laura mentioned it a few moments ago, due to unexpected travel issues with a few of our guests, we had to reschedule the expert dialogue, but we are doing our best to keep it at least tomorrow. In the meantime, we invite you to enjoy your lunch. You have all the rest of us in the university, and we'll join back here at 1 p.m. to continue with, of course, more talks, more learning, and of course, more brain health.